Tes.
Oke, masuk Pak Jeffrey.
Oh, masuk. Oke. 1 menit lagi ya nampaknya.
Rekan-rekan.
Oke. Sudah tepat jam 2 ya. Kita mulai ya. Oke. Good afternoon, ladies and gentlemen. Welcome to the International Webinar Series Fakulty of Farmasi Universitas Sanata Dharma. I'm Jeffrey. It's my pleasure to be your moderator and MC for this webinar series. I'm sorry, I cannot. Yes, series on computer-aided drug discovery and development. This is the first in a series of webinar which organized by Faculty of Farmasi Universitas Sanata Dharma. First of all, I request the Dean of Faculty of Farmasi Universitas Sanata Dharma, Dr. Apoteker Justina Srihartandi, Master of Science, to kindly give an opening speech. Dr. Justina, the floor is yours.
Thank you, Mr. Jeffrey. Good afternoon, ladies and gentlemen. I'm pleased to welcome you to the seminar, which is by Fakultas Farmasi
Sanata Dharma University.
Today we are gathering in the first of 5 seminar series with the topic computer-aided drug discovery and development. Thank you for the sharing.
Thank you for the sharing to Professor Enade and Dr. Maiwan, our lecturers at Fakultas Farmasi Sanata Dharma University. They are expert in this area. Also, thank you to Professor Andreas Bender, our collaborator from Cambridge University.
All of us know
that
very interesting, especially which is related to pandemic COVID-19 situation. Computer will help us to solve some problem, I think, in the drug discovery and development processes. And I think the sharing today will give some idea for us to carry out some research or activity on drug discovery and development. Thank you to all of the committee for preparing and conducting this virtual conference. And also in this virtual conference room, I recognize some familiar names of participants. Thank you for joining this seminar. I'm sorry I couldn't mention you one by one. And enjoying the seminar. And please join with us in the following seminar series conducted by Faculty of Farmasi Sanata Dharma University. I wish you all to get a very fruitful seminar. Thank you, and may God bless all of us. Thank you.
Thank you, Dr. Justina, for the opening speech. Okay, before we start our seminar, okay, we will take a photo session. Please, to all participants to activate the video, and we will take several photos. Okay, please, our team to take the photo. Okay, thank you for the photo session. Okay, we continue to our agenda. Now we get into the first speaker. Again, before the presentation begins, allow me to introduce our speakers. We have Professor Apoteker NAD Perdana Istiastono and Dr. Apoteker Maiwan from Faculty of Farmasi Universitas Sanata Dharma and Professor Andreas Bender from Cambridge University. Our first speaker is Professor Apoteker NAD Perdana Istiastono, PhD. I will read curriculum vitae of Professor NAD. Name Professor NAD Perdana Istiastono, Sarjana Farmasi, Master of Science, PhD Apoteker, address Pogung Kidul, number 7, RT or RW 02049, Sindu Adi, Melati, Sleman, Yogyakarta, Indonesia. For the email address nad.isiastono@gmail.com or nad@usd.ac.id or https://nad.molmod.org. Sex male, place date of birth Madiun, August 6, 1979, nationality Indonesia. Work experience March 1, 2020 until now, professor in pharmaceutical analysis and medical chemistry, Faculty of Farmasi, Universitas Sanata Dharma, Yogyakarta, Indonesia, perform and manage education, research, and community service in the field of pharmaceutical analysis and medicinal chemistry. Okay, this work experience for ancillary activities from 2017 until now, certified research reviewer in Ministry of Research, Technology, and Higher Education of Republic Indonesia, 2014 until now, member of expert committee. Okay, maybe to all our participants, you can read in our screen. Okay, to start. Okay, Professor Apoteker NAD Perdana Istiastono will deliver his presentation with title Protein Ligand Interaction Fingerprinting, the Prediction Quality of Virtual Screening Campaigns. Ladies and gentlemen, please welcome Professor NAD. The floor is yours.
Hello? Is it already?
Okay, Prof. We already here.
Okay, thank you very much, Pak Jeffrey, for the kind introductions, and also thank you very much, committee, for the chance to present here. Let's go to the presentation. Yeah, here you are. Okay. So, can you already see the presentation? Hello? Is it okay?
Okay, Prof.
Okay. The title of the presentation is Protein Ligand Interaction Fingerprinting to Increase the Prediction Quality of Virtual Screening Campaigns. So this is especially made for this occasion of the online seminar held by the Faculty of Pharmacy, Sanata Dharma University. This is the outlines of the presentation today. First introductions, and then I will discuss about pipe leaf, and after that I will discuss about the pipe leaf hippos. In introduction, there are subtopics that I will discuss here: the structure-based virtual screenings and the prediction quality. And then I will close this topic with the molecular interaction fingerprints, or IFP, and employing IFP in structure-based virtual screening campaigns. Virtual screening or virtual screening, or specifically structure-based virtual screening (SPVS), and the drug discovery pipeline here. Here. So the focus of this tool is in this phase. In the after we validated the target, then we screen to find the hits, and after that we can continue to hit to lead and lead optimization drug candidate, and then go to the clinical trial, preclinical and clinical trial. Okay, how I close it? Ah, back up. Nah. Okay, of course, there are many computational things, also structure-based virtual screening, structure-based virtual screening that can be used in the other phases, but I will focus our discussion in the screening to find hit. And why SPVS? Virtual screening also there are some different kind of filter screening, structure-based, at least two structure-based filter screenings, and then ligand-based filter screenings, and also there's some in between the pharmacophore-based filter screening. And then what is computational medicinal chemistry? Yeah, in here I try to define this kind of discipline, subdiscipline actually. It's a branch of chemistry that uses computer simulations to assist in solving medicinal chemical problems. So, and there are some position on this kind of computational medicinal chemistry, closer to the computer or closer to the chemicals, and I choose to be closer to the computers. Therefore, I work with computers to assist. Sorry, so this still, can I erase them? Yeah. Okay. Yeah, so I working closer to the computer. Then my main objective in my research is working with computers to assist medicinal chemists in drug design and discovery. So most topic of my research in our research group is to construct and validate structure-based virtual screenings protocols, and then increase the prediction quality of the protocols. So we will use machine learning, and also we create pipe leaf, etc. So the standing actually here is there's a computer, there are computers. We discuss with computers and we train the computers to give us a more solid hypothesis or more solid suggestions to help the medicinal chemists in drug design and discovery. That's the standing point here, closer to the computers than to the chemicals. If I were closer to the chemicals, then maybe I will prefer to use those kind of validated protocols and then try in silico and then find or optimize some compounds. Okay, can I go next? Next. How to go next? Okay. So why I prefer to choose SBVS? Because the outputs can be analyzed to investigate the molecular determinants in protein ligand bindings. If we understand the lock and key in the protein ligand bindings, aided by computers, it will be very useful in the hits to leads and the lead optimizations. And of course, this is important in fragment-based drug discovery and drug design and discovery. So we will try to maintain the important interactions, and then we will grow up, or we will merge some fragments to have a desired activity. And unfortunately, the structure-based virtual screening much slower compared to the ligand-based virtual screening or pharmaco-based virtual screening that will be presented by Pak Maiwan later. Yeah, I just hope that it could be compensated by the increase of the computer powers nowadays and in the coming days. There are some other reasons why I prefer to use structure-based virtual screenings. One of the reason is based on my experience that using the structure-based virtual screening, we can escape for the similarity with the non-ligand. So because we focus on the structures, the structure of the target molecules, we can escape from the non-ligands. Yeah, then therefore we will gain more novelty in using the structure-based virtual screening compared to this ligand-based virtual screenings. Okay. Now, after I discuss about this structure-based virtual screening, there's some abstractions. Okay. I will now discuss about the prediction quality, because I already mentioned that one of the main objective of the research is to increase the prediction quality. If we construct and optimize, if we construct structure-based virtual screening protocols, we would like to have a protocol that have a high prediction as possible. So there are some objective functions. In the retrospective validations, we would like to have a higher enrichment factor, or F measure value, or F1 value. And of course, we would like to have a prospective validations if it is possible. And then also we would like to have a high resolution model. Since the models after the structure visual screenings could be used for visual inspections in hit to lead and lead optimization. Yeah, then this is the my routine jobs here, doing the retrospective validations and try to have a higher enrichment factor, or F measure, F1 value. And then what is F and F1 value, that we could see the confusion matrix. This is the confusion matrix, the actual class, predicted class. If we use retrospective data, then we already know this is the ligands, and the other way, the rest are decoys. So we will take this as P or positive and this as negative. If the protocols could identify positive as positive, then it is a true positive. If a protocols identify the negative as positive, then this is a false positive. And if the positive identify as negative, this is a false negative, and then the last is the negative identify as negative, this is a true negative. So this is the explanation of those P, TP, etc. And this is the commonly used objective functions, enrichment factor, EF and F1. So it's pretty simple, just percent of true positive divide 2% of false positive, or this F1, the 2 times true positive divided by this equation. True positive plus false positive plus false negative. So in F1, the score will be 0 until 1. The closer to 1 is the better. Okay. This is the commonly used benchmarking set that already provided by Professor Irwin and co-workers. You can go to the website, actually, this duda.docking.org. And we would like to have a SPVS protocol with a bit better EF or F1 compared to the available ones. If you go to targets here, there are several targets. You click each one there, and you will find the EF that they found. It is better to have protocols. If you develop a protocol, you should have a protocol that's better from the one that's already published by DUDA. Or if there's somebody already published the newer one, the better one, so you should have a protocol that's better than the newest one. And unfortunately, there are not all targets that are available there. If there is none, we should attempt to have a better EF or F1 value than the average. So I already calculated the average in the DUDA. The EF is 19.8 and the F1 is 0.207 respectively. So we already covered the SPVS, and then we already covered the prediction quality. And now in 2007, I will discuss here about 2007 technology paper provided by Prof. Marco and Ronyong. They developed a technology called molecular interaction fingerprints, or it's abbreviated to IFP. And actually at that time, these tools focus on fragment, optimizing fragment. And more detail of this kind we will discuss later when we will address this pipe leaf. So in 2007, Marco Ronyong developed IFP, and then we developed here 2013 pipe leaf, and just recently we we published and then we developed the newer version of pipe leaf called pipe leaf hypos. And then I will discuss here about employing this IFP, interaction fingerprint, in SPVS. So the commonly used virtual screening, actually, we just do the retrospective one, do the virtual screenings, and then rank the docking score, and then calculate the EF1. Fortunately, with IFP, we can do some more, and we can rank based, we can re-rank the compound based on the interaction, and we will have a better prediction quality. So in this paper, published in 2015, we show those increase of quality, prediction quality. So in here we use model. So we use several ligand to perform molecular homology model and molecular dynamics. And at that time we have a beta 2 structure as a template and histamine H1 as a template. This is for histamine H4 actually in the model. And we use G&J compound from Johnson & Johnson as a reference, histamines, the endogenous ligand as reference, also compound from Free University as a reference. And here we see in this 4, if we use plants only, the docking software we use, the EF 1% only 2.9. When we use the interaction fingerprint, it increased almost 23, almost 10 times better. And when using this model, the F1% using plants only 1.0, and using plants and IFP, it increased until 17.3. And you can see here, this is the best one actually, from 5.7 until 40.3. And then we use this one actually, these two model to do the prospective validations. So this is first from the zinc database we grab fragment-like compounds and see if there is possibility to have an N positive, and then we perform reactive group filtering using a technique used by provided by Tudor Oprea, and then after that we still get some number 42,000 compounds, and using those two structure-based filter screening, we run them all. We still got some thousands, and we use dissimilarity filtering and visual inspections. Dissimilarity filtering, we use all compounds, all known histamine H4 ligands at that time, and use LBVS actually, LBVS to select the most similar, the most dissimilar. Now after we already get rid of the most dissimilars, and then we do clusterings, and then use visual inspection on those clusters, and then we bought only 37 compounds, and out of these 37 compounds, 9 fragments. Halo?
Ya, oke, Prof. Kedengaran, Prof, suaranya. Oke, we can hear your voice.
Okay, 9 fragments out of 9 fragments of 37 tested compounds. This is represent 5 different scaffolds. Have affinity between 0.14 until something wrong with my sound. Halo?
Yes, halo. Halo, Prof. Okay.
Something wrong with my sound. There's like some kind of feedback here.
Yes.
No, okay.
Okay, it's okay now.
Okay, 0.14 until 6.3 micromolar. So it's very potent. 140 nanomolar, the best at that time. Okay, those experience give the idea actually this IFP, this interaction fingerprint, can really increase the prediction quality, and then we can really use those validated protocols to identify novel novel compounds, not only novel compounds, but novel scaffolds that we can grow to many to yeah, not many more to many more compounds, novel compounds, because we already have a novel scaffolds. And the interesting thing in this experience is we have the similarity bit the interaction fingerprint bit strings, and we can filter out docking poses that do not have or do have a particular interactions. And in this case, in this sorry, in this case, we only take the posts that have a certain interaction with the aspartic acid 332. So this is the nice thing that we learn from this interaction fingerprint, that we can filter out based on a certain interaction. Therefore, in 2012, after I finished my PhD, I tried to figure out how to have this kind of technology here. So this is the next topic, the pipe leaf. I will discuss about pipe leaf development, and then I will discuss about the structure-based virtual screening using pipe leaf, and then structure-based virtual screening using pipe leaf and machine learning. Pipe leaf. Yeah, as I told you previously, that this is inspired by Marco Rognong AFP, which is C++, and I don't know, perhaps it can only be used in Unix kind of operating system. And the software uses the OpenAI libraries, which is proprietary, and therefore we try to have this pipe leaf. This is a Python-based protein ligand interaction fingerprinting. We use open bubble libraries, which is with free and open access libraries, and we choose Python implementations. So this is kind of basis for the operating system cross-platform. So we hope that we can develop more to be used in other operating system like Windows. And yeah, since we worked a lot with the plants docking software, the pipe leaf at that time, using output from plants docking software as the input. So this exclusively developed for plants. Actually, we can work around that using a simple scripts, but I won't discuss here. Okay, this is plants. We are after docking with plants, we have this process, and based on the process, we go to pipe leaf, and then pipe leaf will identify the 7 interaction. Diapolar interaction, aromatic phase to phase, aromatic S to phase, S-bond hydrogen bond interaction with protein S donor, S-bond interaction with protein S acceptor, ionic interaction with the protein positively charged, and electron interaction when protein as negatively charged. So this is the output of pipe leaf 1010. So we see that it is kind of hacking the compound and change those two into a bit string. This is the example of a pipe leaf output here. And this rule, actually, we have a rule. We use a rule base stated by Marco Rognong. And then we tried to use in a structure-based virtual screening in estrogen receptor alpha. So if we use plants, CAMP-LP, the docking score of plants, the EF already high, 21.2. If use the scoring function from the pipe leaf, the TC leaf, it's a little bit decreased from 21 to 17. But because we have the bit string, then we can filter out some filter out compounds without these interactions. So we only take the compounds that the poses that have this kind of interactions. The EF 1% increase significantly, become 53.8. Unfortunately, this kind of interaction, this kind of increase was chosen without the previous info, without previous information. So in the histamine H4, we use information from a site-directed mutagenesis study, and then we thought at that time there's possibility for some favorable interaction or non-favorable interactions in certain target, but we do not have the knowledge of that. We really need to dig the library, the reference, to find the important interactions. Or we can also do the systematic filtering from the sports bit string to identify important interactions. Okay. The systematic filtering, we tried using this histone deacetylase as the target. Yeah, it increased actually. We found that this is the important bit string interaction, number 117. Thanks to Dr. Nunung Yuniarti and Dr. Arif Nur Rahmat that provide me with the in vitro data. And then see if we use the default, the EF is only 15. If we use the TC flip, it's also still very low, 1. But if we use the filterings, if you use CAMPLP is increased, the TC flip is also increased. So the main factor that increase the prediction quality is the filtering on the bit string 117. This value outperformed the EF value of the reference protocol from Meisinger et al. Okay, if you use TCIP or TC flip, unfortunately, those are reference dependent scoring functions. This also experiences on the structure-based virtual screening in the histamine H4. If you use different reference, and then the quality, the prediction quality is different. And then, how about we use flip bit string with machine learning? And then at that time, I discussed with Prof. Iwan DS at Pre University. The preferred decision tree is the preferred machine learning technique is the decision tree actually, because we we can also identify the important interactions, not only a black box. And at that time, I tried to search in R computational statistical programming, and we found a tool called RPart. This is a recursive partitions and regression tree. And then this is the first trial using RPart, still using the estrogen receptor alpha. This is the result. So using this RPart techniques, we will also identify which bit string is important. So if we follow the path, then we will find whether it is will designated or predicted as decoy or ligand. And then, yeah, it really increased the EF value. Really increased the EF value. So see, it's the reference value is 18.5. We use the best decision tree. It's increased until 200, almost 250. And also we also identify the important residue and the interaction type. But some weird things we recognize here, like is it possible that glycine provides a hydrogen bond? And the answer is possible. The backbones. So also this the leucine can make a hydrogen bonds. Yeah, the answer is possible in the hydrogen bonds. And that and then we try actually with using difficult targets, XCR4 did not work, the decision tree did not work well, and we derive from bit string into an ensemble brief. So from page briefing, we derive using a simple algorithm to create an ensemble brief that we abbreviated into ENSPLIF. We published in 2017. The F1 become 0.58 and the EF is 323, is very high. And this also we try again in ER alpha. This is the algorithm to create the ensemble brief. So this is the common algorithm, the preparations, the dockings, and in the docking we have n number of poses, and there's a bit string. This is the bit string from the pipe brief. And then we just calculate the sum of this interaction divided by the number of poses, and we will have the ENSPLIF value. And then we use this is the we thought ENSPLIF, and this is the one that using ENSPLIF. So this is and then okay, perhaps this kind of a lock and key theory. And then the key is not rigid, it's a flexible, and the lock is also not rigid, it's a flexible, and then we should use the induced fit theory. So this is the solder. If we illustration for a key, the key number 1, this is the shoulder. KMPLP should be less than minus 60, and this is the cut, first cut, and split number 320, etc. So this, with using this and split, we can increase the value. So this is the SPPS2. The F measure increase dramatically actually from 0.1 become 0.7, and the EF also increase significantly. The SPPS2 is using the normal flip, not the end split. It's slightly increased. And then we also use this kind of techniques for another target. We choose here the acetylcholine esterase. We have when we perform the retrospective virtual screening using SPVS plus pipe leaf, and we calculate end split, and using air part, we can have a protocol that have a F1 value of 0.4, still below 0.5, but is still acceptable. And this is the interactions and the residue discovered by this guy with these techniques. And interestingly, most of them are hydrophobic interactions. And we apply this validated SPVS on peptides. So this is the first paper using the in here we present that as natural product, and we discover for about 30 compounds from natural product that are predicted as positive. And then we want to do VS on peptides, and this is the site results. If you want to screen peptides, then we should have the peptides. Then then we construct the peptide first, and we have a bunch of almost 80,000 compounds, 80,000 peptides plus already, and we upload in the database that are publicly available. And using those, we screen those peptides, the dipeptides, 3 peptides screened, and none of them are active using this protocol, this validated protocol. And then we screen selective tetrapeptide. We found 4 compounds, and then we design based on our knowledge in the first paper. We find the AEUTR, alanine, glutamate, tyrosine, threonine, and arginine, the pentapeptide, as a potential acetylcholine esterase inhibitor. And 2 more to go actually, that this active peptides, the predicted active peptides were synthesized and tested. And then we found that those compounds are comparable to donepezil or even better affinity to acetylcholinesterase inhibitor. Donepezil is the ACE inhibitor in the market for Alzheimer. Okay, and then we thought about improvement during this experience. During the use of pipe leaf, we know something that lack from pipe leaf. It only accept output from plants. Interestingly, we know that some of the people in this field know that now the Vina, autodoc Vina have a growing popularity. And pipe leaf calculate reference bit string in every run. And pipe leaf, there is no option to avoid interaction to backbone atoms in pipe leaf. Therefore, we can identify here glycine, leucine have a hydrogen bond because the hydrogen bonds goes to the backbone atom. Therefore, recently we published the newer version of pipe leaf. We call it pipe leaf hippos. Pipe leaf hippos stand for hippos is pipe leaf on steroid. It's like a recursive abbreviation actually. Like Vina, autodoc Vina, Vina is not autodoc. And inspired by those, we use a pipe leaf hippos. Hippos is pipe leaf on steroids. And it just published 2 or 3 months ago actually. And this new pipe leaf is not only exclusive for plants, but also it can receive input output from autodoc Vina and go to pipe leaf hippos. This is the icon of pipe leaf hippos. Cute, isn't it? Like hippos. And then we will have the fingerprint bit string. The new feature of pipe leaf hippos actually it accept also output from autodoc Vina as the input, not only from plants. These new tools also 10 times faster compared to pipe leaf. Why? Because it separates reference leaf calculations. There's also option using no reference if you don't want to compare to any reference. And these tools also directly assign atom type based on the used docking software. Oops. So pipe leaf use rely on Open Babel to assign the atom types. And there's also option no BB, no backbone, to avoid identify interaction to backbone. And also, if you want to have a similar different similarity coefficient, IFP only provide the Tanimoto coefficients, and then pipe leaf also Tanimoto coefficients, and there are many other similarity coefficient of bit string. Then we provide options to modify the similarity coefficients. The default we give here is the Tanimoto similarity and McConaughey similarity. Okay, then next, in my next projects, I would like to implement of lock and key theory and industry theory in SPVS, and then use those kind of new theory, new tools, or new approach to make a highly predictive SPVS protocols and more user friendly. Actually, I already prepared demo, but unfortunately, during the rehearsal this morning, that the demo could not be done. Then perhaps I will try to make some demo and then put it online in YouTube or in some other occasion. Thank you very much. I think this is it. Time and place for you, Pak Jeffrey.
Yes. Thank you very much, Professor Enade, for an interesting presentation. Now we get into the second speaker. Second speaker is Dr. Maiwan. Dr. Maiwan Ariono will deliver his presentation with title Structure-Based and Ligand-Based Pharmacophore in Drug Design and Discovery. Before Dr. Maiwan begins his presentation, I will read curriculum vitae from Dr. Maiwan. Full name, Apotheker Maiwan Haryono, PhD, email address mharyono@usd.ac.id, work experience as lecturer at Faculty of Farmasi, Universitas Sanata Dharma Indonesia, education, S1 from Farmasi Universitas Sanata Dharma, S2 from Ilmu Farmasi Universitas Gajah Mada, S3 from Kimia Farmasi University Science Malaysia. Here several selected publications in prestigious journal. Achievement, Timur 1 Perakmi Award 2017, Indonesia Tore Science Foundation Award 2017. Address Jalan Sabu, Sopalan, Maguwoharjo, Depok, Sleman, Yogyakarta, Indonesia. Okay, ladies and gentlemen, please welcome Dr. Maiwan. Dr. Maiwan, the floor is yours.
Thank you, Pak Jeffrey, for the kind introductions. My gratitude also to Prof. Andreas Bender and Prof. Enade, to all speakers, and of course to all participants, ladies and gentlemen. Okay, without further delay, I would like to share my presentation. Okay. Okay, I hope that you can see clearly. Sorry. Yes. Okay. Today I will talk about structure-based and ligand-based pharmacophore in drug design and discovery. If previously Prof. Enade talked more about structure-based drug design, which is used a lot in the virtual screening to find hits, for the drug candidate. But if the condition is there is no protein structure, the 3D protein structure available, so we have to think another way around to design the drug, which is based on the ligand. So the structure base, it's actually both are structure, but the term of structure base belong to the structure of the protein. But for the ligand base, it belongs to the structure of the ligand on the chemical compounds. Yeah, so I will start with the definition of the pharmacophore. Yeah, according to the very famous microbiologist and also the medicinal chemist, Prof. Paul Ehrlich, that pharmacophore is a certain chemical groups or functions in a molecule were responsible for a biological effect, and molecules with similar effect have similar functions in common. Yeah, for example, this is the structure of morphine. Morphine is in pharmacy or in a medical is very famous for a strong analgesic. We can see that morphine is composed by phenol group, phenol structure, which is hydroxy group attached to the benzene ring or phenyl ring. And also the tertiary amine, nitrogen with 3 hands or 3 bonds is a tertiary amine. And also ether is the oxygen bridge. And the last one is the alcoholic, hydroxyalcoholic, close to the cyclohexene ring. So based on the Paul Ehrlich definition, it's still concerned on the chemical groups, functional group. It's the past definition, yeah, that is responsible to a biology. So actually not all structure in drug will responsible to the biological or pharmacological activity, but a certain, so for example, like this phenol, only amine, only allylic alcohol, or only ether that will make an interaction with the biological target. Other definition from chemobiodynamic and drug design by Schuller is pharmacophore is defined as a molecular framework that carries, because in the Latin, foros means carries, and the essential features responsible for drugs, or in Latin language is a pharmakon, biological activity. So it's a backbone or a skeleton that carries features or character or properties that will interact with the biological target, leading to the pharmacological activity as a drug activity. But this definition is no longer concerned. It's not about the chemical groups, but actually is about the pattern, yeah, about the pattern, the system of how the space of the chemical groups or the functional groups position one and each other. It's about the distance and also the angle each chemical groups or functional groups. And it's highlighted also by the IUPAC that a pharmacophore is the ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interaction with specific biological target and to trigger or block its biological response. So this is the ensemble, it's the combination of steric. Steric means voluminous, yeah, because when we talk about the interaction between drug and the target, is about the 3D and the 3D space, not like what we see in the whiteboard. So it's about the steric, the volume, also the kind of electrostatic character of between the ligand and also the biological target to make interactions. Yeah, maybe we can illustrate with morphine first, that as introduced before, that morphine has a kind of functional group which is important for interaction with the specific target as an analgesic to the opiate receptor. So this is a phenol, and this is the tertiary amine or nitrogen. So it has a character as an aromatic. This is the can be hydrogen bond donor also, as well as the hydrogen bond acceptor. But the nitrogen with 3 bonds will has a character of a positive or a cation. So it could be make an electrostatic interaction with the biological target. So this is the hydrogen bond, and this is the van der Waals or hydrophobic interaction with the biological target. So from the whole structure of protein, actually it's not important. Yeah, the most important part of the morphine is only for the phenol group and the nitrogen. We can modify anything in here, yeah, not always, because the function of this kind of yellow backbone is only to support to make the position of the phenol and the nitrogen is stable, is there and is there. So this actually is just for the supportive, not for the specific interaction with the biological target. So we can actually just remove the yellow backbone, and we found the most important structure in the morphine to act as a strong analgesic or painkiller. So if we compare with other strong energetic, which is the morphine derivatives, for example, is metazosine and liforphanol, we can see that actually for the strong energetic, the thing we need just for the phenol and the tertiary amine. Yeah, we can see that the almost same thing in the morphine derivative is there is phenol and tertiary amine. Here also, the difference is the supporting backbone. Yeah, we can see that no matter what the structure of the yellow of the supporting backbone, but the most important thing that phenol and tertiary amine should be there. Okay, so we can just reconfirm that actually pharmacophores is define the relative position of the functional group. So it's not about the kind of functional group only, but also the position. The phenol should be there, the nitrogen should be there. So this distance and also this angle must be specific, must be certain. Cannot just you put the phenol is here and then the nitrogen is there. So cannot. That pharmacophore is not just a kind of functional group, but also the relative space. Yeah, also the distance included the angle. So yeah, this is better explained here that the position of phenol and nitrogen is expressed by the distance x in angstrom, for example, and then also the angle here. The angle is must be specific. If we just change a bit about the angle, it could be inactive, yeah, not active as a drug anymore. If we change the either the distance as well as either the distance or the angle or both. Yeah. So this is the important talking about. So back to morphine. Yeah, as we know that in drug discovery, we have been exciting with the in silico method, yeah, as previously elaborated by Prof. Enade. One of kind is the structure based, and then this one is the ligand based. That we can also use in silico method or computational method to generate a pharmacophore or to identify actually which part of the ligand is important for the binding interaction with the protein target. So the computer will convert the 2D structure into the 3D one. Yeah, and then the computer, the program used to generate pharmacophore will define the kind of functional group and also automatically measure the distance as well as the angle. So we can see that actually the morphine here is kind of three ankles, yeah, three ankles with a specific three ankles and the distance from the O is from the OH, yeah. So AR is from the aromatic ring or benzene, and then N nitrogen is from the tertiary amine. So based on the knowledge in the chemistry, that O is could be a hydrogen bond donor as well as hydrogen bond acceptor, and then aromatic process the non-polar interaction, hydrophobic interaction, something like that. And then the nitrogen also could be, if there is a positive charge of cation, so it could possess the electrostatic interaction with the binding site. So from the program, they will define that the pharmacophore of strong analgesic, which bound to the opiate receptor for analgesic, should be this distance, and this distance. And also this ankle, and this ankle. Yeah. If we just shift this ankle to this side or this location, so it could be not working anymore as a strong analgesic. That's what pharmacophore is talking about actually. So yeah, so define the relative position which will interact with the binding target for the pharmacological activity. We have to know about the hydrogen bond acceptor, what kind of chemical group is categorized as a SBA. Yeah, we abbreviate it as a SBA. The example for the SBA like CO, carbonyl, yeah, oxygen have a lone pair electron, so it can accept hydrogen from other. In contrast, hydrogen bond donor, yeah, it should be rich with hydrogen, so it can donate the hydrogen to other which needs the hydrogen. So van der Waals interaction is talking about the hydrophobic London dipole-induced dipole that you have to recall about the basic chemistry about the chemical interaction. And last but not least is ionic interaction is also sometimes is very important to possess the biological activity for the ligand. Okay, so this back to the pharmacophore of morphine, that morphine should have a SBA here, van der Waals interaction here, and the ionic interaction here. Yeah, so we can confirm, yeah, the conformation is very important. Yeah, so actually that position, that relative space between in between the functional group is very important for their stability. Once the drug bind to the receptor or to the binding site of the protein target, they should be not just bind and then leave. Yeah, so we can imagine for the drug that first time give the effect, what we call in pharmacology science is onset, yeah, but the duration is very short. So could be the drug has no time to cure the patient, but the drug is already eliminated or extracted from our body fluids. So the stability of the ligands, it's just not about the energy of binding, which show you a stronger interaction, but also the stability, yeah, stability. So usually pharmacophore also consider about the conformational analysis, identifies possible conformation and their stability. Yeah. Also, conformational analysis is difficult for flexible molecules. Yeah. That's what happened when we have a drug candidate having a lot of rotatable or flexible as can be assigned from the single bond. Yeah, when the drug have, that's why when we open the pharmacopoeia, yeah, pharmacopoeia or pharmacopoeia Indonesia, that we saw the structure is on benzene, benzene, benzene. All the drug have the benzene because the most favorable conformation by the drug is rigid. Yeah, it's will reduce, yeah, it will reduce the instability of the drug if the drugs most of the drug has a rigid structure. For example, is we can see from the dopamine. Dopamine is one of a neurotransmitter in our brain. So the dopamine basically have a benzene with two hydroxy in ortho position, one each other, and the meta position from one of OH group having ethyl bridge and also ended with the amine group. So dopamine is not stable because it has two single bonds here, which is rotatable. So other analogs, drug designer will think about how to reduce this rotatable, because rotatable makes the conformation is unstable. So they put the lock the chain here to be the ring, yeah, from the chain to be the ring. So it's kind of more rigid and not moving anymore, not rotatable anymore. And the other analog also, but the position is here. So actually, from the aspect of the pharmacophore, we can see that people want to reduce the instability of first compound or lead compound to be better stability, in the better stability by locking the structure. But we have to think about the pharmacophore. If we modify the structure, the pharmacophore is there. Yeah, still we can see it's the same one. The distance from OH and to amine group is same. In the first analog also same. From here is also same. But we can see that the position of OH group and H2 here is different. So we can shortly conclude that it could be, yeah, it could be the first analog will as active as dopamine, but it's more stable. In the second analog, it could be stable, but maybe it's not as active as dopamine because the pharmacophore is not same. Yeah, we are talking about the relative position of one and other functional group. So yeah, we can just imagine from the binding site in which the binding site show you an important amino acid residue, something like aspartic acid with CO2 minus. It has a acidic character and also negative character. And then serine here with OH, good hydrogen bond donor or hydrogen bond acceptor, and phenylalanine here with a phenyl ring acting as a hydrophobic pi-pi interaction, that kind of. So if we want to create a ligand based on this important site, if we want to interact with negative, we have to put the positive or the basic one. Because this one is acidic and negative, we have to design drug which can interact. Yeah? Negative will interact with the positive, acidic will interact with the basic. And also, if we saw here serine with OH, we have also to put the H bond donor or H bond receptor, because OH can donor the hydrogen as well as can accept the hydrogen for others. So you can put here either SBA or SBD. At this site with phenylalanine aromatic center, we have to put simply just put the same thing we can put here is benzene. Yeah. So actually, from this pharmacophore, we can define that the pharmacophore for this binding site should have basic or positive center, aromatic center, and H bond donor or acceptor. Then we can create the triangle, something like that. That's a pharmacophore, not just what kind of function, but also the relative position. And then, other things, again, pharmacophore triangle from the dopamine, the pharmacophore dopamine is here should be SBA, SBD, SB, SBD, aromatic, and also the basic or positive character from the NH2. Then we can create the pharmacophore or dopamine is here. But later in the computational, it automatically will be generated by the program or software of the computer. This is just an exercise how to identify the pharmacophore for the oxamniquine is for the treatment of schistosomiasis. If we have 3 analogs here, so we can identify actually which functional group as well as its relative position will be make a sense to be the pharmacophore of the treatment in the schistosomiasis. Now, as we can see that here the point is in the NHN. So in the same position, para position with methyl and methylene, CH2 here, along with the benzene ring here. So we can say that the pharmacophore is here actually. Other is just supporting. It's just supporting. So what is the pharmacophore functional group commonly named or commonly categorized or classified? So we can classify that common pharmacophore features are its bond acceptor. For example, its carbonyl group. In software, it can be colored by what kind of color? Cyan, cyan. And then it's BD donor with a magenta, and then the functional group having an anion negative character would be colored as red, and then for cationic would be blue, and then hydrophobic is green, and then the aromatic is brown. Here is the software will identify the kind of pharmacophore. What is BD, and then hydrophobic, aromatic. There is a benzene ring, and then could be the here, magenta is SBD, and then the relative position is certain. Cannot just we can freely make position according just to a kind of function. No, it's about the pattern on the relative position. So the software will create the pharmacophore. So the pharmacophore is here. So we can see that the spheres, green spheres, is kind according to the, for example, hydrogen bond acceptor, it should be here. And then hydrophobic, the blue one, it should be here. This is the cationic, and then this is the SBA anymore. So you can use this pharmacophore queries as the filter. Yeah, we can filter. We can make a virtual screening as like Prof. Enade talked before, using structure-based virtual screening. By using the pharmacophore, if we have a big database, chemical structures, so we can use the pharmacophore to filter. This pharmacophore is already defined as the functional group position that will be active to interact with the biological target. So the database will be screened out with the pharmacophore filter, and then we'll found the hits. Yeah, the hits will be identified. So from, for example, from 10,000 of chemical compounds from the database, we will just select 20 or maybe 100, compounds that score that have the highest score after we filter. So it will be ranked. Yeah, the top score will be selected as the hits. So this is actually the scenario if we want to design, if we want to do the drug design. Yeah, this is the metric. Yeah, in the left side, if the protein structure is yes, means that it's available, as the protein is not available, and in the top side is ligand is known and no, yes. Yeah, so we can choose the scenario according to what we need and also what the situation is. So if the protein no and the ligand no, so it's a magic. It's just a magic. You just can hope the lucky, yeah, or the serendipity that by the magic you can create the drug. Yeah, we thought protein structure is available, neither the protein nor the ligand available. So it's a magic, but maybe it can do. So the witch doctor, tabib or dukun, yeah, there is no knowledge about the protein and ligand, but sometime they can make the patient relief from the disease. Who knows? We don't know. Then if the protein is available, and then the structure is available, we can do docking. Yeah, because docking needs both. You need the ligands and as well as the protein. If the protein available but the ligand is available, yeah, we can do what kind of a de novo design. Maybe you can just find first what is the definition. De novo is we can identify the part of, or like what we have done. I think this one is de novo. Jadi, so we have to know the important point of the binding site. Then we can imagine what kind of ligand should be post there. Yeah, post there. So that's the condition when the protein is available but not for ligand. And when the protein no, protein yes, yeah. When the ligand yes and then the protein no, sorry, this one is we can do the ligand-based drug design, something like pharmacophore, QSA. Yeah, so the focus is on the ligand and their experimental bioactivity. Yeah. But if we have both protein and ligand, so we can do the structure-based like molecular docking or molecular dynamics. Yeah, this what we did talk about the de novo design. So based on the binding site, we can create, we can imagine, and then put our idea on the computer. So the computer will agree not to create the pharmacophore as our idea. This is one of software to generate pharmacophore. Could be structure-based drug pharmacophore, or ligand-based pharmacophore, yeah, ligand scout from interligand, pharmacophore queries built from features of a protein site. So we just input the structure of protein of interest complex with the ligand or inhibitor or agonist or antagonist. So we just can input the ligand scout, and the ligand scout will easily, very simply identify the part of the pharmacophore of this kind of ligand. Yeah, then these features describe the principal interaction between the protein and its ligands. From the software, we can see what kind of interaction between the ligand and then the amino acid residues surrounds. Yeah, we saw the 2D visualizations. Yeah, so like this carboxylate group, yeah, carboxylic acid group will interact with electrostatic or hydrogen bond with, for example, aspartic acid, tryptophan, lysine, and also the aromatic ring here, signed by yellow color, will interact mostly with non-polar interaction, hydrophobic interaction, phenylalanine, valine, isoleucine, and valine, and so on. Then LigandSlog is notable as the first software package ever to construct automatically a query from one or more protein data bank file based on the protein ligand interaction. Yeah, this is the visualization when LigandScout identified just put the file from the protein data bank we have downloaded, and then just press the button create pharmacophore, and it's very simple. LigandScout provides the following pharmacophore. Maybe from among one and other software will have a different color for its common pharmacophore features. But in LigandScout, the hydrogen bond is assigned as a green color, acceptor as a pink color, hydrophobic with yellow, and then this kind of flowers with a blue with positive ionizable area, negative ionizable area. It's like a donut, yeah, aromatic ring. Then excluded volume is the inaccessible space. So the functional group or the structure of ligand could not reach that kind of position is defined as an excluded volume. Yeah, this LigandScout can identify the pharmacophore based on the distance constraints, represent the relation between 2 points, one located on the ligand side, one of the macromolecular, so from the ligand and the protein itself. So, for example, aromatic interaction with positive ionizable will be identified at 3.5 to 5.5 angstrom. Yeah, its bond interaction from 2.2, 3.8 and so will be identified as its bond interaction, and so on. Yeah, and this is the after we uploaded the structure of protein complex with the ligand. Yeah, so it's directly retrieving PDB complexes online. This task can be performed by using the dedicated control panel or PDB panel. So means we can type the PDB code. Yeah, so the software itself will automatically search for that kind of PDB code, and then will be automatically uploaded in the display on the visualize of the LigandScout. So this PDB panel displays important information on the current macromolecule ligand complex, provide instant PDB access, and is composed of 3 components. Yeah. PDB 4-letter code instance, you can type 1KE7. So just press, and then automatically it will be uploaded in the display. Yeah. Or we have to download it first from the PDB, and then just file open something simply like what we open the file from some software. Yeah. Then the macromolecule view provides structural information on the macromolecule and the ligand's position with it. So we can see that this is the protein. Yeah, this is the protein, and then this is the ligand. It's highlighted with a yellow box here. Usually we just can click the box and it will automatically zoom it. And we can see the position of the kind of ligand is very clearly. Yeah, active site view displays structural information on the ligand and its essential surrounding macromolar environment. If we explore, yeah, we just can rotate and explore actually what is the surrounding amino acid residue, what kind of the ligand, yeah, what is common features is there. So yeah, we can have an idea what the software will predict as we our idea has been raised before. So creating structure-based pharmacophore is we have to import the data, structure preparation, then the software will automatically generate the structure-based pharmacophore. Yeah, it's kind of like just import the selected PDB file here, like type 1KE7, and then press, and then it will be uploaded in the display. Yeah, alternatively, you may use the create pharma. Then there is a pharmacophore button here. Yeah, pharmacophore button. Then we just press, and then ligand score will calculate the pharmacophore model within seconds. It's very quick. So back to that's about the structure-based pharmacophore ligand using ligand scout. Yeah, another way around to create pharmacophore also using ligand base. So there is no protein available. We just have a set of ligands. Probably minimum is 16. Yeah, 16 to 20 that we can create the trend or the linear regression. Yeah, linear regression. Like we are doing calibration curve when we use a UV spectrophotometer. Yeah. So if the ligand known and the protein structure known is structure based. Yeah. If the ligand known but protein unknown, we can do QSA, pharmacophore alignment. If the ligand structure is unknown but the ligand known, we can the ligand unknown, the protein structure is unknown, is known as the de novo design. And the last one is the if both unknown, yeah, could be a magic, or we just can do what they can do, yeah, library design, searching similarity searching, or analysis diversity. So what is the QSAR talking about? It's a quantitative structure activity relationships. It addresses two questions. What features of a molecule affect its activity? What can be modified to enhance the property? So at the first time in the QSAR, we will to know and define what is the features that makes that molecule or ligand to be active as a drug. Then after we get an idea, we have to create or we have to design one or more molecule that will be fit into that model that we have created before. So it helps to gain insight into underlying biological processes. Yeah. So also it alleviates the need to determine the experimental value, that the property of hundreds silicon that would take large amounts of resources to determine individually. It means that nowadays, yeah, we do not need to synthesize or to isolate a lot of compounds. Then we have to test to know which one is better or not. But from the previous data set, we can get the equation, yeah, simply set as a linear regression equation. Yeah, so that equation we can predict a new compound or a new ligand that we don't know its activity to. Then after we put or apply this chemical structure of the unknown ligand to this equation, we can predict the activity before we test by in vitro or in vivo. So it's QSAR is a mathematical relationship between a set of physicochemical properties known as descriptors. Descriptor is actually same with the pharmacophore name as features, but in QSAR it names as descriptors, but it's actually is more like these things. And the bioactivity on the set molecules. Yeah. So from this scheme, descriptor is a molecular structure alongside with the target activity. So from the chemical, we have to do the bioassay test, then we got the activity, and from the structure and the bioactivity, we can create mathematical equation, or we can transform it into a mathematical equation. Then we can use this equation, yeah, generated equation, to predict the activity of the new compounds. So like, yeah, we know this kind of um, multiple linear regression actually, because it's kind of why that we have been familiar with this, yeah, the x and then the what we call slope gradients and then the intercept and something like that. But this equation, mathematical equation, is a function actually. It's a function of the biological activity with the chemical descriptors, the descriptor. For example, this is the molecular volume, and then log P, we know log P is the ratio of the concentration of drug in the what we call a coil of N-octanol, and then over by water, and other descriptors, something like SBA, SBD, and so on. Yeah. So objective is to correlate Y with X1, X2. Yeah. So this could be if the positive character, so the PKI, PKI is kind of like IC50. Yeah, IC50 is the activity of the compound is as a Y, and then these descriptors as the dependent variable. Yeah, dependent variable. So variation is one dependent. So variation in the X1, X2 that are correlated with change in bioactivity and make sense. That's the QSA talking about. Yeah, this one are the physicochemical properties on the descriptors, such as hydrophobicity of molecule, the whole molecule, and the hydrophobicity of the chemical functional group, substituents, yeah, electrolytic property of substituent, and steric. Yeah, steric is the following most common property studied. Yeah, it's done. And then, yeah, so if the protein is not known, is unknown, yeah, but the ligand known, if we have a set as ligand, we can design the pharmacophore based on the structure of ligands, not from the protein. So a set of multiple active and inactive compound may be known, and more advanced pharmacophore modeling can be utilized to create pharmacophore. Yeah, so first, the conformational space of flexible molecules need, which we have talked about, what needs to be covered extensively since the bioactive conformation are unknown. And then second, the molecules need to be aligned. Yeah. So the software will align the pharmacophore with the common pharmacophore features. It can be retained in a 3D model, something like this. Yeah. So from this two compounds, that actually two compounds is different. You can see that it's different structure, but it might be having a same pharmacophore, because even though it's different structure, but here the white one has a carbonyl, and then the green one also has the carbonyl at the same position. So it could be different structure, but have the same pharmacophore. You can see also here, the nitrogen here at this space area, at this position. So it could be having the same pharmacophore. So software with the ligand-based pharmacophore can identify that. Yeah. Using inactive derivatives, the essence of the feature as well as the permitted steric arrangements of the ligands can be mapped as well. And then, yeah, the algorithm used in the BioVia Discovery Studio software is named as a catalyst hypogen. In particular, stands out from the variety of tools available for this purpose. Yeah, this is a combination of QSA and the pharmacophore method. Yeah. It attempts to correlate structure and activity values, Ki or half maximal inhibitory concentration, or we knew as a IC50, by constructing a pharmacophore model. So if we have a set of structure and then the activity provided by the bioassay, so we can create a pharmacophore according to the QSA. It's combination between QSA and the pharmacophore. Yeah, by using QSA, then the software will identify actually which chemical functional group will important for the activity. Yeah. Then creating ligand-based pharmacophore. Yeah, diverse chemical structure compounds along with the corresponding, as I told before, IC50 or Ki value ranging between potent, yeah, the IC50 should be very low, and then moderate, yeah, could be in 1 to 10 micromolar, and inactive is more than 10 micromolar. Yeah, we can classify which compound is classified as a potent, moderate, inactive. Yeah, to create pharmacophore, these three categories should be there. Yeah, should be there. Then 3D star pharmacophore, yeah, can be used to generate predictive pharmacophores, and then the catalyst hypogen algorithm to derive SAR, structure activity relationship hypothesis. So it will create not only one, usually is depend on our request. Yeah, if we instruct, yeah, and we instruct to the program to create 10 pharmacophores, then the software will consider, yeah, it's reasonable or not to create pharmacophore or less. Maybe it depends on the parameter that, and also the set of the ligands we give to the program. The descriptors are concerned with the overall molecule rather than a single substituent. So it's about the pattern, not a single substituent. Yeah. Generates the diffuser models that are based on how well a series of ligand fit to pharmacophore. Yeah, so if we can fit the compound to the pharmacophores already created by a set of compounds, then should be the activity can be predicted, yeah, before we synthesize or isolate, and then also before we do the bioassay. Yeah, this using Biofra Discovery Studio, the catalyst hypogen algorithm undergoes 3 steps for creating pharmacophore. First is constructive, followed by subtractive and optimization. Constructive means common pharmacofunctional alignments of the most active compounds, training compounds will be generated. So at the first step, it will identify the common conformational alignments. By using this formula, it's to identify the most active compound. The formula is set as the most active compound MACT times uncertain MACT subtracted by the ACT, the training set compound activity we studied, over uncertain activity should be more than 0. Then it will be defined if the compound, one compound fulfill this equation, then it will be assigned by the software as a most active compound. Secondly, the subtractive phase. Catalyst will remove some hypotheses that fit inactive compounds from the training sets because we want to identify the active pharmacophore. So the one which show log ACT in bracket subtracted by the logarithmic of the most active one in bracket should be greater than 3.5. So it will be assigned or it will be identified as inactive. Then this kind of features will be removed automatically by the catalyst. And then the last one is the optimization phase. Yeah. Catalysts employ fine perturbation in which adding or removing new feature to chosen hypothesis then survive from the subtractive phase to find new models of improved bioactivity and mapping correlation. This is using statistics. It will be optimized which pharmacophore will be chosen for the virtual screening. So the application is virtual screening approach for TMPK inhibitor in a TB, yeah, for TBC, tuberculosis, to identify the drug that should active for TB. So this is the outline, pharmacophore generation, stored before, selection of hypothesis, and then the virtual screening. So virtual screening, we create the pharmacophore from the 88 compounds with the known activity. Yeah. Later, I can maybe show you how to use, but this having a license, so we cannot just freely using this kind of software. Yeah, so basically it's to create the pharmacophore, and then after we select by statistic, we have to choose 2 or more pharmacophore which make a sense to use this as a filter for virtual screening. And then this pharmacophore will be used as a filter to screen compounds like this and identify the hits. For example, compound 5 has a fit value score better than others, the best one. So we can predict this compound would be active by in vitro. So it's the visualization on how the compound 5 map to this kind of pharmacophore in also the second hypothesis. Yeah, and then will be validated by the ROC, receiver operating system, considering the decoy as Prof. Enade talked before, yeah, for the external validation to see the enrichment factor. It's actually the protocol of virtual screening using pharmacophore is valid or not. Yeah, this is just how to define the ROC. Basically, if the ROC is a plot about the sensitivity for the true positive rate and false negative, false positive rates, if the area is tends to the upside,
Dr.
it will be
Maiwan, I'm
okay.
sorry, due to your
Yes.
time is
Yeah.
up.
Thank you. It's just
Would you mind to jump to the last
okay. Thank you, Pak.
slide or conclusion?
It's done. It's done. Yes.
Oh, it's done.
Yeah.
Okay.
It's just a kind of a publication I have done doing pharmacophore modeling, and the other one. Thank you, Pak Jeffrey. I return
Yes.
the times to you.
Yes. Thank you very much, Dr. Maiwan, for an interesting presentation. Pak Mai, I'm sorry. Would you mind to stop share
Yeah.
screen?
I'm trying, but don't know how to do. Maybe I just close my file from Windows.
Okay. Thank you.
Stop. Is it okay, Pak Jeffrey?
It's okay, Pak Mai. Thank you. Yes. Now we get into the last speaker. The last speaker is Professor Andreas Bender. He will deliver his presentation with title. Oh, I'm sorry. With title Artificial Intelligence in Drug Discovery and Pitfalls. First of all, I will read the Professor Andreas curriculum vitae. Dr. Andreas Bender is a reader for molecular informatics with the Center for Molecular Science Informatics at the Department of Chemistry of the University of Cambridge, leading group of about 22 postdocs, PhD, and graduate students, and academic visitors. Maybe you can read the Professor Bender CV from the slide. Okay. I will continue to the due to Professor Bender unable to join live in our seminar, so we will watch his presentation recording. We apologize for this situation. Ladies and gentlemen, we will start to play the recording video of Professor Bender's presentation. Please.
Yeah, hello and welcome everyone. And thanks a lot, Inado, for inviting me to present at this conference. My apologies that I cannot be present in person. It's Saturday, so it's a weekend here, unfortunately, so I need to do something with my family. But hence I wanted to record this presentation at least and send it over to be present at least virtually. So what I will present is the use of artificial intelligence in drug design. And I want to take a slightly critical, slightly reviewing angle to this type of information. Right now, AI is hyped quite a lot in many areas, and there are great successes. For example, when it comes to image recognition, when it comes to speech recognition. However, in the drug discovery area, applications of machine learning and AI are unfortunately not yet bringing the success that we see in other areas. So I would like to ask the question why that is the case. And as we will see, there are reasons in particular when it comes to the underlying data. For example, biology is very difficult to deal with, and we will review that a little bit. But not everything is lost, of course. We have lots of data, chemical data, biological data. And so we want to and we need to analyze that data and to make good use of that. And there's still certain value in doing that. So I will give some examples of that as well. So I would like to start my talk, please. So the title of my talk is Artificial Intelligence in Drug Design. What is realistic? What are illusions? And so first I should mention that this is an academic talk. So I also started companies, for example. I'm working for AstraZeneca, but I'm also an academic at the University of Cambridge. So what I say today, I'm speaking purely in my capacity as an academic, and so it's my personal opinion only. So what is the situation when it comes to AI and drug discovery today? There are many things happening. And so these are some of the headlines I was capturing that appeared in the recent months and years. And so we see some of the headlines: artificial intelligence is changing drug discovery, first breakthrough in AI drug discovery, and so on. And there are certainly lots of things happening. And you see new success stories, and that is great. That is great because there's really quite a lot of effort of making use of chemical and biological information. So that is great. However, despite all that effort that gets put into that area, we see relatively few successes. And one of the reasons might be that drug discovery takes quite long, let's say 10 years on average. If you go from the discovery of a hit through all the different phases to determine efficacy and safety of a drug, and then to really get the approval of a drug on the market. That process takes long. Clinical studies in particular can take years. It depends on the indication. It costs a lot of money. You have cohorts of 1,000 or so patients in some cases in order to show efficacy and safety of a new drug. So that requires quite a lot of time and effort. So it might simply take so long. But on the other hand, there are also conceptual reasons why things don't work out so well, as we might see on the following slides. So one of the advantages of getting older is you recognize certain patterns again. So I'm old enough to remember the 2000 biotech bubble, for example. So I was working at a cheminformatics startup here in Berlin. And so I was a summer intern. So I was a student who was working for the first time implementing cheminformatics algorithms nearly 21 years ago now. So I remember that. I remember the Human Genome Project. Human genome was sequenced. There were papers, big papers, published next to each other in the reputed journals, and there was quite a lot of hope behind that. But let's revisit that. So in 2001, there was a review article that commented on the Human Genome Project, and what that article says is the number of drug targets will increase by at least an order of magnitude, so tenfold, and target validation will become a high throughput process. So that was in 2001. And there will be more drug targets, so 3,000 to 10,000 targets, compared with 483. So that was the number that you found in review articles. I believe it was a Nature Reviews drug discovery article as well, that determined that was the approximate number of drug targets at that point in time. But recent, so 2017, estimates of drug targets put that number at 667. So we see it's a larger number, so we have 200 new targets, 1/3 more. It's quite some progress. But certainly in 2001, that number, it was a bit overhyped. We didn't get to 3,000 to 10,000 targets. It's simply not the case. So sometimes if you're in the middle of a new initiative, of a new trend, it's easy to be optimistic, and it's very good to be optimistic, but it's easy to get carried away with that a little bit and to say that you found the solution of it all. And sometimes that is simply not the case. So what we will review in this talk is the following. So we'll go through the current data landscape, so chemical and biological data in particular. We'll talk about deep learning. So that's one of the methods that rose in popularity quite a lot. So some of you might have seen those papers that computers are beating Go masters, for example, image recognition, speech recognition. There's great progress. But we'll talk about chemical and biological data as well as, for example, biology, and that is closely linked. So deep learning, you need lots of data for that. And quite often we don't have that, and we might not have the right types of data. So I will go to biology afterwards. And finally, I will conclude with the interface of AI, human psychology, and society. Because if you have trends in society, those trends, they're usually not only determined by pure science, they're usually determined by additional human factors at the same time. So we'll talk about this interface a little bit as well. So this is a slide I was usually using to explain to groups what we are doing in our research group at the University of Cambridge. So this is the data we look at. That's the main data types, I should say, that we look at. So we look at molecular structure, we're looking at proteins or mode of action, as well as phenotypic information. And in some sense, that's quite a comprehensive view, because the molecular structure, that is chemical information, this is something we can modify. We can synthesize very different drugs, or compounds, I should say. A drug is a compound that's approved on the market. So we can synthesize lots of compounds, many of which are bioactive. That's kind of the independent variable in our system. So that gives us quite a lot of power. We can make nearly any compound we want to make. And from that molecular structure, we have links to the protein, the mode of action. And that's, for example, bioactivity data. So if we think about PubChem, we think about Campbell, we have millions of data points that link molecular structure to a particular protein, or more generally, mode of action. Because you can have direct interactions, so structure interacts with a protein, or you can have indirect interactions. So structure binds to the aryl hydrocarbon receptor, for example, that induces expression of certain cytochrome P450s. Okay, so that's an indirect target. So you have biological signaling cascades. But in both cases, what we have is a link between a molecular structure and a mechanistic level of compound action. And we also have links between molecular structure as well as phenotypic endpoints. And that's usually, if you think about drug discovery, what we're actually interested in. So we want to kill a cancer cell, for example. We want to inhibit replication of a virus, and so on. So this is what we observe on a macroscopic, on the endpoint level. And all of those links are actually supported by lots of data. So we have the molecular structure that is linked to proteins, a mode of action, for example, via data from PubChem or Campbell. Then we have pathway annotations. So this is information that connects different proteins or genes in a systematic manner and links them to phenotypes. So we have, for example, a signaling cascade that links a particular collection of kinases to the cell cycle, for example, or the growth inhibition of tumors. So we don't have independent genes or proteins, but they assemble in a certain organization. But I should say, humans like to think in pathways and categorize things. In biology, of course, nature is not like that. So it's maybe better to think in terms of a network. And that network only dynamically reconfigures. You have a different cell type, you have cell division and so on. So those pathways are activated, inactivated. Certain genes are expressed, others are not. So that is very dynamic. So maybe our viewpoint of thinking in terms of annotated pathways in a binary way, it's probably simplified. But still, pathways help us organize biological information. And we also have links between molecular structure and phenotype. And that is in some sense the most difficult type of data to deal with. And so that is data that is very dependent on the particular setup of an assay. So if, for example, the phenotype is the observation of an adverse drug reaction, then this depends heavily on the genetic setup, the dose you take, for example, the age, the sex of the patient, is there any co-medication, is there any other disease that patient has, and so on. That is a very tricky angle of this data triangle pyramid. But the point here is we have lots of information that links structure to mode of action as well as a phenotype, and all that information we can mine. And we can usually ask 3 different types of questions. So if we have the molecular structure and we have the protein, the mode of action, then we can use that to design, to anticipate which phenotypic changes occur. So for example, we inhibit, I should go to this slide. So if we have, for example, information about the molecular structure as well as the molecular target, then we can predict which patient, for example, responds to a particular drug. So how do we do that? We know, for example, a particular viral protease, so that's the mode of action level, is inhibited by a particular drug. And if we then determine what the genotype of a certain virus is in a given patient, then we can say what is the right drug for that particular patient, and we can say whether the viral replication gets inhibited or not. So starting from the bottom of that list here, in this case, So the question is, and that goes into the area of personalized medicine, which patient responds better to a drug? That works very well, for example, in case of HIV, where it's easy to sequence viral genomes, costs only a few dollars right now, and where there's lots of experience, large databases, which particular virus gets inhibited by a particular drug. So that's the personalized medicine angle. But if we go to the top of this list, we can also ask the question, if we have the molecular structure, so that was at the top of this pyramid, and we have the phenotype, so we, for example, observe a adverse drug reaction, we can ask the question, what are the proteins that are associated with that particular event? So what are the proteins that cause a particular adverse drug reaction? So in this particular case, we would ask for the mode of action. So we can do something like target prediction. That was an area of heavy research in our group. And finally, if we have the link between a particular disease and pathways that are involved in that disease, upregulation of pathways, for example, that tells us which proteins we should inhibit, kinases in the signaling cascade, for example, and that tells us then in turn, via the data we have, which chemistry, which focused library we should make in order to have compounds that are pathway focused, so chemistry that acts on kinases in a particular pathway to achieve a desired effect. Okay, so these are three different questions. So usually we have information about two of those angles from the pyramid, and we ask, and we infer based on data, we ask a question about the third corner. Okay, so it's very powerful. But as you might have seen in the previous slide already, this is the world is flat view. And what do I mean by that? This whole view is quite simplified, right? Because if you're familiar with drug discovery a little bit, you'll know that in order for a structure to have an effect and cause a phenotype, for example, a compound needs to have the right distribution in the body. It shouldn't be metabolized or cleared too quickly, right? So there are lots of types of information missing in that triangle. And that's very important to keep in mind because this is basically the problem why artificial intelligence approaches are difficult to apply to drug discovery data. It's very conditional. Yeah. So the labels are not absolute. If you have a molecular structure, you cannot say that drug causes drug-induced liver injury, for example. It depends on the dose, the genotype, and the sex, age. It depends on lots of things, many of which we don't even know. So it's not binary annotations. And that is what makes data mining and drug discovery space very difficult. Yeah, the world is not flat. So what do we mean by that? There was only one example. Some other reasons why the world is not flat, or data mining and drug discovery space is not so easy. Some of the other reasons are that links between drugs, targets, and diseases are quantitative and incompletely characterized. So what we don't know, for example, is if a compound is a partial agonist or full agonist, characterizing pharmacology is difficult. Then we have subtle differences in compounds' effects. Different types of agonism is actually quite you don't only have activators and inactivators, right? You can have allosteric modulators and so on. You can have off targets, residence times, so kinetics of binding can be different. You can also have different downstream signaling, for example. So you have biased signaling. Yeah, so in a G protein coupled receptor, you can signal via the G protein or beta arrestin and so on. So it always depends on what happens in a particular system. It depends on which other receptors are present. You have kinetic effects and all that. It's very difficult to characterize completely. It's very difficult to understand what a compound does in an in vivo system. Yeah, it's relatively easy to do that in an isolated receptor assay, but once you have a huge biological system, many receptors expressed, you have kinetic effects, different distribution in the body, that's very difficult to do. And then effects are state dependent, so we have a variation between individuals. For example, it even matters what you've eaten in the morning, whether a drug works or not, because that changes the PK profile, the uptake of a compound. And that's simply often not captured in the data we have. Yeah. So we measure in an isolated cell assay. So what does that mean for humans? And that in vitro to in vivo extrapolation, that's something that's very difficult to do. And in particular, when it comes to data, phenotyping is sparse and very subjective. So if you think, for example, about diseases, sometimes you have the same genotype, but in two different individuals, it is apparent on the phenotypic level to a very different extent. So why is that? What's the reason for that? Are there other mutations, for example, polymorphisms in the genome? Are there other reasons for that? Or is it something else that mediates that effect? Yeah, and phenotyping is difficult. So, for example, different doctors have a different viewpoint on a particular observation. If you have a particular symptom, for example, that means something different in the context of a different disease, in the context of other symptoms. Yeah, so that is very subjective and very difficult to do. You can also see a disease on many different levels. You can say that there's a cause of the disease and there's a particular mechanism and there are the symptoms. Yeah? And sometimes those levels don't completely agree with each other. And so this type of data is very difficult to handle. And finally, we don't properly understand biology, so we don't know what to measure and label. So we can measure easily gene expression, for example, but why is it actually gene expression? One reason, of course, is microarrays became cheap, for example. And of course, genes are quite essential, but we know that the gene level and the protein level are only very weakly correlated. And then proteins can be phosphorylated, activated. So the question is, which level, gene level, protein level, metabolome level, phosphoprotein level, and so on, actually gives us the information we need. Quite often we don't know that. So we're generating data on a very broad scale, but the question is, where's the signal? Yeah, and quite often I believe this data or noise to signal trade-off is not very favorable, so it's a bit more on the noise side. We generate lots of data that don't tell us anything. So going from data to information and understanding is very difficult. And the type of data we have really depends on the context. So if you think about that in the drug discovery situation, there are two very different types of environments. And I would call that right now usually early discovery as well as late stage drug discovery or safety. So what does it look like in early discovery? So that's more hit identification, for example, high throughput screening, proxy assays. That's the area we operate in here. So quite often here we have simple readouts. So for example, activity on a protein. And that means it's easy to measure. So we have a large number of data points, which is good for training models. And models have clear labels. So for example, we measure agonism against receptor A. And so we have a nearly completely defined system within biological variation, but for example, levels of receptor expression and so on might vary, but we have a biologically relatively defined system where we can easily measure large numbers of data points. And those data points are many data points and they have clear labels. Yeah, so we can say the ligand is active against the protein A, with an IC50 below 10 micromolar. Same is true for log D as relatively defined system. And all of that is very good for model generation. That is easily mineable data. But if we go to in vivo biology, and that is what really matters in drug discovery, of course, the situation is quite different. Because in this case, we have quantitative data, so things depend on dose, exposure, right? If you take 10 times the amount of a drug, then things look different. That is difficult to encode in models. It's another dimension. And it depends on time, for example, as well. Yeah, so along the time axis, you have effects. So how do you encode that if you do data mining? And so what we have in this case is for data generation, we have more complex models to generate data and also fuzzy labels. So classes depend in the simplest case on dose, for example, but they also depend on your assay system. Or if you're in vivo, they depend on the particular biological setup of a certain patient and so on. Yeah, so we have fuzzy labels, and that's difficult for data mining. In one case, on the left-hand side, we can say compound is soluble. On the right-hand side, whether compound is toxic depends, right? It's conditional labeling. That's difficult to mine because you have lots of conditions you need to distinguish. So in this case, in the in vivo setting, we have less and less clearly labeled data, and that's difficult from the machine learning angle. And the same applies not only for labeling data, it's even recording data that is tricky. So imagine you do animal studies. It even matters whether you put a cage at the bottom of the floor or at the top, because then light is different, and also fear levels are different. For example, if you're at the floor of the handler of the animal is male or female, because stress levels are higher if handlers are male, for example. And all those are confounding factors in your data. That is much more apparent in late stage quantitative data, but unfortunately that data is much more in vivo relevant, okay? So there's a trade-off between having proxy data and lots of it, but the extrapolation to in vivo is less clear. So what does an IC50 against the isolated protein target mean in vivo? On the other hand, we have late stage data, in vivo relevant, but it's much less data and it's fuzzy labels, yeah, so it's more difficult to handle. And so basically it's a trade-off, right? We don't have a perfect world here, yeah, so it's a trade-off, one versus the other. And of course, also the question we ask the data is quite different. So the problem setting in early discovery versus safety is different. In early discovery, usually we want to find some solutions out of many. So we have 10 to the 60 molecules we can make. So if we have a screening library, we have 1 million or so compounds. And then the question is, give me a set of suitable few dozen, hundreds, maybe thousands of compounds in the screening setting. And it's my enzyme below 10 micromolar or so. That will be something I could call a hit, and I can continue. So we're predicting here the presence of something. Predicting in the computational sense, or otherwise measuring the presence of something, but it's a setting where you want to discover something out of many possible solutions. And that means from the computational side, generative models, for example, are often useful. That's one of the big directions of machine learning models right now. On the other hand, in the late stage and safety area, we need to predict for a particular data point. Yeah, so there's not the option for the model to say, okay, I don't know anything about those compounds, yeah, which is possible in the early discovery setting. We will identify some examples, and the false negatives, so to speak, we don't care about. In this particular case, we need to predict for a particular data point. So the model needs to be accurate for the compound we ask it to make a prediction for. And that is much more demanding for a model because it cannot focus on the area it knows about, basically. And also, there's usually a large number of criteria to rule out. So, for example, many modes of toxicity. So you're predicting the absence of many things based on limited data, based on a small number of data points that is difficult to label. Okay, so you see on the left-hand side, discovery setting, we have more data, it's more easily labeled, and it's an easier question we ask the computational model. In the late-stage setting, we have less data, it's more fuzzily labeled, and we're asking a more difficult question. Okay, so it's quite a difficult setup on the right-hand side. And predictive models, they are more tricky than generative. For example, data coverage is limiting. So we see, I mean, if we go to the in vivo setting, real-world situations, then the data we have is limited in amounts. The labeling is more difficult, and we're asking more difficult question. Okay, so early discovery is easier in this sense. So discovering a ligand is many times easier than discovering a drug, for example. So, but not everything is lost. So we can and should still use the data that we have. So I'd like to give a few examples of that. So the first example is target prediction. So that's one of the case studies I was mentioning before. So in this particular case, we have a molecular structure, we see on the top, and that is linked to a phenotype. So we have a particular drug that shows an effect, phenotypic effect, based on data. So a drug kills a tumor, or cancer cell in the simplest case, or it causes an adverse drug reaction. And based on that structure and lots of data, we're able to predict the protein or the mode of action. And so there are public models available for that. So one of them was constructed by a then PhD student in my group, Lewis Mervyn, who is now with AstraZeneca, and he was also a student with AstraZeneca. And so that is a public model where you can just put in a structure and you can then predict protein targets of that particular small molecule. And that is publicly available in case you're interested. And so this is a very important use, and it's one that works reasonably well, in my opinion, where we can do data mining. We can use, if you wish, AI or machine learning approaches in order to make sense of chemical and biological data. So the model can predict protein targets, so putative modes of action of a compound. And that is, in my opinion, relatively possible. So what do I mean by that? On-target activities, so links between chemical structure and protein targets, are quite data rich. So if you think about Campbell, PubChem, and so on, we have thousands of data points for many targets, GPCRs and so on, enzymes in many cases, and relatively homogeneous. Yeah, so that falls onto the left-hand side of those categories we had before. And hence, generating models for on-target bioactivities is generally possible. It's in quotation marks because it depends on how many data points you have. If you, for example, have one very large class and a small class, it's difficult that the model really predicts the small class as well as the large class. You have lots of biases and so on. So, but still, overall, the data is relatively homogeneous. We have, relatively speaking, lots of data, so we can generate models relatively easily. And it can also be used for design. So in this case, it was used for explaining effects. You have a compound linked to an effect, but we can also use it the other way around. We can say, give me chemistry that binds to those two targets, and then we can design a new structure. But there are also limitations, of course, and they are very important to keep in mind, and only covers known biological space. So the models that we build and that are based on data, they're also limited by the data. So we use the data we have, but on the other hand, it's difficult to extrapolate to completely new areas of chemical space. And that depends a bit on descriptors. So the question is, how do you capture the chemistry, the information you have? You want to put it on a level where you generalize information. So use pharmacophores, for example. There are different ways to do that. But generally, still, models are always limited by the data you put in. They also suffer from various data biases. So sometimes you have large classes, you have small classes, more diverse classes, less diverse classes. So normalizing the model output is not trivial. Labels are still heterogeneous. So if you imagine if a kinase inhibition assay, that depends on the ATP concentration, which particular output value, IC50 value, you're measuring, for example. Yeah, so there's still heterogeneity in that system. If you have a radioligand binding assay, it depends on the radioligand, which particular activity value you get, whether you bind exactly with the radioligand binds, so you have a competition, you displace that ligand or not, and so on. And of course, in vivo relevance needs to be established. So that means if you have a target prediction model, you put in a compound, you predict a certain target to be bound by the ligand. That only means that potentially, if the compound reaches the protein, it binds to it, but does not tell you whether the compound in vivo reaches the target, and does not tell you whether it reaches the target at the desired level. Yeah, so the desired concentration, so that the IC50 value of that compound actually matters in vivo. And does that, thirdly, not tell you whether that is responsible for the effect. Okay, so just predicting a target does not mean you know the mode of action. You only have a hypothesis for a mode of action that you can test. So this is a slide that probably most people will disagree with in one way or the other. But the point here is, so what is plotted here is the attention received in the recent past on the x-axis. So how many publications do you have in those areas? And on the y-axis, whether, in my personal opinion, that scientific question is solved. And what you see here is you have, for example, toxicity and efficacy prediction, so predicting in vivo endpoints. And they are quite large, and that means that's the impact on the drug discovery process. So they have lots of potential impact. If you predict toxicity and efficacy, you can predict whether you have a drug. But there's little attention because of lack of data and so on. And the question is certainly not solved either. Okay, so it's a question that is very important to answer, but it's difficult to answer. And it didn't receive much attention due to lack of data. Then repurposing, for example, here you're staying in the in vivo situation. You're using a drug for one indication for another. It received quite a lot of attention in the recent past. And in some sense, the question as well, solved. We know some approaches how to do that, whether on a target level, using omics data and so on. But it's not truly solved because it doesn't work for any mode of action. If we don't understand the disease, we also don't know which data to use for repurposing. If you go to the right, for example, in the top right-hand corner, we see some topics that are shown in blue, and blue means they are on the chemical domain. De novo design of compounds for targets, reaction prediction, target prediction. So they received lots of attention. The scientific questions, well, I'm not quite sure they are solved, but there certainly has been lots of progress in those areas. But unfortunately, the impact on drug to patient is, relatively speaking, a bit lower. The reason is that they stay in the chemical domain. If you want to develop a drug, you don't have a compound for a ligand for a target. You need to have overall favorable properties related to toxicity and efficacy. Okay, so you can interpret that figure in a way that you say models go where the data is. The data is on this left-hand side of the model generation pipeline that I described before. Yeah, we have simple proxy assays and lots of data points, and this is quite often the case in the more chemical domains. We have large databases with reaction schemes, for example, which are used now to predict the forward and backward synthesis of compounds. We have large databases with bioactivity data, so we can do target prediction and so on. Databases, and they're difficult to construct as well, that tell us whether a compound is efficacious, because there's a huge number of parameters that plays into that. Okay, so basically AI and drug discovery quite often goes where the data is, and the data is where the proxy assays are, the simple data points are, but they're quite often less in vivo relevant. Okay, and so what I believe is to make more of the AI approaches that we have in the drug discovery context, we need to go more into the biological, but more difficult areas as well for predicting toxicity and efficacy. Yeah, and so the reason for that is biological data is painful. So what do I mean by that? So if you're a data scientist, you might ask the question, so does the drug cause an adverse reaction or not? So you want to label the drug with the adverse reaction. Drug A, aspirin, causes stomach bleeding. And by and large, that's true. It happens quite frequently. But does it do that in every case? Of course not. Not in every patient, not at every dose, and so on. Okay. So this simple labeling is not possible. So if you ask someone from the pharmacovigilance department, that person will respond, well, it's not so easy. If you have a patient with this genotype, usually unknown, who has this disease endotype, insufficiently defined, who takes dose X of drug Y, but sometimes also forgets to take it, so that means you don't really know actually what the dose is or the exposure to the drug is, then we see adverse drug reaction set, but only in 12% of all cases, and only if co-administered with a drug from class C, and then only in males and after a certain time and so on. Okay, so this illustrates biological data depends on many conditional variables. Okay, so it's difficult to analyze this type of data. It's very difficult to make sense of that. And the analogous, of course, holds for cellular systems. So you have cell line drift. Even if you have very simple cytotoxicity assays in cell lines, you have mutations. There was a recent Nature paper on that, 2019 or 2018. So even if you have cell lines that are ordered from a supplier and that are characterized, you have mutations that occur. So the cell line is not the same in every instance, and that's a huge problem. Because if you think about even simple cytotoxicity data, if the cell lines are not identified properly, that is a big problem. Then you don't even know which system you measure in. That's a problem, right? The same is even more extreme for animal histopathology data, where actually the assessment of organ slices, for example, is very subjective. Okay, so biological data is truly painful. That's one of the reasons why in drug discovery, many people shy away from that. Yeah, deep learning. So that was one of the big trends over the last 5 years or so. So in my opinion, it can work well. And sometimes it works well numerically, but it doesn't really address the underlying question. And thirdly, it's sometimes pushed in a biased way in publications. And I will give examples for each of those, but it certainly does make sense. But it works numerically well, and that's one of our own studies. But the question is, what is the endpoint you model? And that is very important, needs to make sense. And thirdly, sometimes the way results are presented is also a bit biased. So where can deep learning work well? And so it can, in the area of target prediction, I think it can work quite well. So there's a recent paper by Andy Meyer, large-scale comparison of machine learning methods for drug target prediction on Campbell. And so these are, that's the essential figure from their publication. And you see that neural networks outperform in this particular case all other methods. And so you see that on a large benchmark data set, you get good performance from deep learning models. Of course, then one question is it take the computational time, parameter optimization, and so on worth it? Potentially, depends on the case. And very important, statistical significance is one thing, but does it translate into practical relevance? That's very important to distinguish. Significance and relevance are two different things. If you, for example, have large numbers of data points, you can have a tiny effect size, so tiny average difference between performance, for example, but because you have many data points, you're still significant, but it can be practically meaningless, okay? So always discriminate both things, statistical significance and practical relevance. They are, by and large, independent. So you can have, in some cases, even practical relevance without statistical significance. If you have small sample sizes and you have a trend, yeah, that can still tell you what is a better model to use. But, and that's the important part, statistical significance does not always mean practical relevance. So mentally keep both of them a little bit apart. And that's a reference of slides that you might want to look up as well in case of interest. So that's the first case. That's where deep learning doesn't make sense, I think so. The second case is a study we did ourselves. Is on modeling synergy of anti-cancer compounds using deep learning. So synergy is in many cases desired. In cancer, for example, infectious diseases. So if you take two drugs instead of one, you can lower the dose, but they still, because of synergy, so super additive effects, they still have a considerable efficacy in a particular patient. In some cases, it's not desired though, because imagine that the compound shows synergy against a particular cancer, that cancer mutates, yeah, and that means if it mutates in a way that synergy decreases, the cancer is able to escape the mutational pressure. right? The pressure of the drug. So basically, the drug combination is less efficacious if the cancer mutates. Okay, so that means you're actually driving the cancer away from the situation that your drugs are efficacious. Which means if your drug combination is antagonistic and the cancer mutates, you actually capture it because the drug combination becomes more active against the cancer with the mutation. So don't assume synergy is always desirable. I guess that's my point. Always depends on the case. And there were recent combination screening data sets published by Merck, AstraZeneca, NCI, and so on, which we can use to model synergy. Synergy, so super additivity, seems like a valuable thing to model. That's what we thought. That's what in some sense I still think it's just difficult to do in practice. So I would like to self-critically evaluate our work. So does this matter, synergy modeling on cancer cell lines in practice in the real world? So firstly, that's what we did. So we had deep neural network. So we have descriptors for drug A, descriptors for drug B, and we have the genetic data, gene expression, for example, of the cell line. And then we can, depending on a combination of A and B and the genetic data, predict the synergy score. And we found numerically, if you take a deep neural network compared to some other approaches, we actually can predict synergy better. Okay, sounds good so far, right? Numbers are better. But is that really the case? So let's have a look at the numbers first of all. So we have a synergy score of 30 as a threshold, so significant synergy of a combination of two compounds in a given cell line. And we get a true positive rate of 0.55, true negative rate of 0.95, means effectively 1 out of 2 positive synergistic predictions is correct on average, while 19 out of 20 non-synergistic predictions are also correct and can be rightly discarded when looking for synergistic compound combinations. All right, so a true positive rate is about 0.5 and true negative rate is 19 out of 20, so this is where those numbers exist. But what's the practical relevance? So synergy is dose dependent, and here it's measured in cancer cell lines. So how does it translate to the in vivo situation? It also depends on the particular definition of synergy, because you need to assume what is your baseline model, your null hypothesis. Do you assume that two compounds, if you add them together, how do you define additivity of particular of the compound combination? Because you can assume if the compounds are, for example, located in the same or different pathways, you have different underlying hypotheses, what your additivity hypothesis is. And this means you have a different baseline, so you define additivity in different ways. Yeah, you have the HSA, high single agent model, and various other Lowy model and so on. They all make different assumptions, which means synergy is not a property you can define only in one way. And the same is actually true for many endpoints we have. If you think about solubility, for example, sounds quite simple, right? But you can measure it in many different ways. You can have thermodynamic solubility, kinetic solubility, and so on. So it's many of the endpoints we measure are more difficult to measure than you think. And here, synergy, there's even a subjective element to it. The subjective element is how we define additivity. Yeah, but the big question here is, it's dose dependent. How does it translate to in vivo? Yeah, and that is a question that is not answered at all in this work, but it's practically very relevant. Yeah, so we can have a machine learning model, produce good numbers, but the question is, if you want to do drug discovery, how do you use it? This is something that is not addressed in this work, and that's generally difficult to go from the in vitro to the in vivo situation. Okay, so this is an example where you get better numbers using deep learning, but there's still the question of how do you translate that into practice. Yeah, and the third case is, sometimes work is also reported in a biased way. And for example, there was this paper, Scalable and Accurate Deep Learning with Electronic Health Records, received quite a lot of attention. And so they were predicting mortality and so on using different methods. And in the abstract, they say deep learning models achieved a high accuracy for tasks such as predicting in-hospital mortality, readmission, length of stay, and so on. And here you see the area under the receiver operating characteristic curve. So the OROC scores marked in different colors. So this is the performance for the deep learning model. And in the supplementary material, last page, somewhere hidden, they compared the deep learning model to a baseline model, logistic regression. And they said for the full feature enhanced baseline and so on, they measured the same things with this baseline model. And so the corresponding numbers are shown here in red. And you see that the deep learning model has an OROC value of 0.93 to 0.94. And that's pretty much what you get for your baseline model as well. Okay, so the the abstract as well as the title imply, deep learning outperforms so many other methods. But actually, that's not really true. Within the confidence interval, the baseline model and the deep learning model are basically show the same performance. Okay, so always check if there's a baseline model provided in a study, and see if statistically and also with respect to effect size, a model that claims to be superior really is superior. That is not always the case, as this example illustrates. Yeah. But are there still cases where we can use biological data? Yeah, I believe so. For example, omics data is often difficult to model, but it certainly can contain a signal. For example, for repurposing, and we've done that quite extensively. We also started a company, Helix, it's now 60 or 70 people in Cambridge, based on that principle. So you can use gene expression data, for example, for repurposing, finding a new indication for a drug. If you go back to the question of early stage and late stage modeling, we needed to distinguish two different types of modeling. One is one out of many selections or definite predictions for a given molecule. In this particular case, we're looking for new pairs of drugs with indications. Yeah, so there are many possibilities. And if we don't require to repurpose a particular drug or find a drug for a particular indication, then we actually have lots of options we can pick from. And that is quite beneficial for the selection. It increases our success chances. In our experience, transcriptomics data, so gene expression data, often contains a sufficient signal for signal detection, but possibly less so for modeling. Dose response is difficult, for example, and hence also extrapolation in vitro to in vivo and so on. And so this is at the bottom, that's my tongue-in-cheek but rough guideline. 1/3 of the time nothing happens in gene expression space if you apply a compound. 1/3 of the time too much happens, you don't understand the signal, and 1/3 of the time you see something you can use. Though you might have already known this before anyway, but you see that omics data is not applicable in every case. It's only if you hit a transcription factor or signaling cascade that you see meaningful signal. So it always depends. But in many cases, empirically, it's easy to generate. Affymetric strips only cost a few £100. You can measure very shallow, you can measure gene expression levels for a few dollars now, depending on which technology you use, TempoSeq and so on. So you can measure that quite easily. So it's a very good signal to cost trade-off, in my opinion. And where we use that is, for example, for the selection of compounds to induce stem cell differentiation. So you have stem cells, you put on a compound, and then you have a protocol for 3 weeks that differentiates the stem cells into cardiomyocytes. So this is the cells in your heart that beat, that contract. And what you see is that after 21 days, if you look at it through a microscope, that you really see a beating cardiomyocyte. That's quite impressive. That really shows me how powerful computational analysis methods are. Yeah, you pick a small molecule which you put onto a cell, and then you can distinguish, you can influence the development of that particular cell and differentiate into tissues. It's also important for pancreatic beta cells, for example. If you get older, people develop diabetes, so you can differentiate stem cells in your pancreas. So to differentiate new stem cells into pancreatic beta cells, for example, regenerative medicine in the eye. We're getting older, all of us. And so the question is how to regenerate tissues, and small molecules more easily reach various parts of the body. So that might be one approach to differentiate stem cells. And so in this particular case, the computer, based on gene expression data, picked a small molecule that differentiates the stem cell into cardiomyocyte. That's a really fascinating study, in my opinion. It's one of my favorite papers from my group, I should say. Yeah, and so I would like to conclude with a discussion of the data we have, technical problems with model generation, and also the intersections of AI with being human and society. So the data we have, much of that data, I would think, is generated for the wrong reasons on wrong ways. So what do I mean by that? Quite often it's proxy measures to reduce cost. So for example, we would in principle like to know whether a compound reaches a particular target organ. That is very difficult to measure. So we measure to characterize a compound and its properties, solubility, log D, let's say, some permeability of CACO2 cells or something like that. And we try to infer from this type of information what the pharmacokinetic profile of a compound is. But that is difficult to do. because the in vivo system is more complex. And so in practice, quite often proxy measures are used to reduce cost. So we generate lots of data points in model systems, but then it's difficult to say what that means for the in vivo situation. It can be an irrelevant system, dose or time point. Quite often it's hypothesis-free data generation. So we basically profile a compound in a gene expression array. We don't know what we're looking for, and then we have lots of parameters, variables that change genes. Then the question is, so what do we do with that? And sometimes very subjective to analyze high-dimensional data, and that can be tricky. And that's one other learning, I think, for me. Try to do hypothesis-driven research. If it's hypothesis-free, you generate lots of data, but the question is, where's the signal? That can be very difficult to figure out. If you have a hypothesis, this gene matters in this pathway, yeah, as a marker for a certain disease, and measure that, you have only a single variable, a few variables to look at, instead of 23,000 or so. Now, in the bigger picture, that means that quite often there's a technology push and not a science pull. What I mean by that is there's a technology, sequencing, that gets cheaper and cheaper. Then the question is, so what can we sequence? Yeah? So that's not the driver. We need the sequence in order to do something. There's the driver. Technology gets cheap, so this is what we should do. This technology push. And sometimes that's not the right way to do things, because you want to have a science pull so that you generate the right data for the right purpose. And that's discussed in more detail in this blog post here. And you can summarize that in this particular way. That's what I realized is very important to think about scientific data in this way. So first comes the question. So not the data, first comes the question. So we need to identify, so which parameters matter in our particular system. Do we see a signal on the gene expression level, phosphorylation level? Which pathways do we need to look at? Yeah, because that determines which data we need to generate. So then we have, that means that we have data that's relevant for the question. We need to generate it in the right amount and the right quality, number of replicas and so on. And only then, once we have the data, the question is, how do we represent it? So think about it, for example, bioactivity data, how do we represent our molecules? Do we summarize gene expression data and pathways and so on? So these are representation questions. And only on top comes the method that captures relevant relationships. And that can be a machine learning method, AI method. And right now there's lots of focus on the method. How do I analyze data? But basically my point is, the question comes first, then I need to have the right data, I need to represent it in the right way, and only then, at the top, the model can change things. But it depends on all of the layers below, okay? So it's very important to understand the question, the area you deal with, and that the data is generated in the right way. Yeah? It all starts from there. Don't start with a method. Ask yourself the question, is the hypothesis proper? Is it the right data for the right purpose? Do I represent things in the right way? And only then comes the choice of the method. Yeah, so in other words, a method cannot save an unsuitable representation, which cannot remedy irrelevant data for an ill-thought-through question. Okay, so start at the base of this pyramid. Yeah, sometimes it's very difficult to actually ascribe performance or success to a method in the machine learning sense. So if a drug results from the drug discovery pipeline, it's not as linear as the term implies, so that's why it's in quotation marks. It's the result of a long series of choices. So basically, if there's a claim that AI discovers a new drug, well, I mean, what is responsible? It's impossible to say because, and you can see it in different ways. On the one hand, there might not be a baseline for control. So usually if you validate a method, right, you need to have a baseline, a negative and positive control ideally. Sometimes you only have a baseline method, so negative control you would probably call it, and then you compare it to your method. But then you should not do a proof by example, so only test one compound each. You need to have a large enough number of compounds suggested by one method and by another method. And then you need to compare, I test 50 compounds here, 50 here, here 12 are active, here 23 are active, okay? So that means the success rate of my new method is much higher, okay? And quite often it's proof by example that is done. And if my computational algorithm discovers a certain drug, if it would do that, didn't happen yet, I should say, but then one viewpoint is we don't have a baseline, okay? It's proof by example. On the other hand, the viewpoint B is, but it worked. Look at the compound. What I mean by that is, in drug discovery, there's always a subjective element. Humans pick a compound over another because of gut feeling, okay? So there's always a subjective choice, and you can have both viewpoints, and sometimes they're both valid. Scientifically speaking, we don't have a baseline. It's true in many cases, or the sampling is not large enough. But on the other hand, if things work, they work. Yeah, that's a justification as well. I think both viewpoints have some value, but to quantitatively compare models, probably it's good to have a baseline and do large enough prospective validations. But that's hardly done because, of course, you don't want to test as many compounds for your baseline or negative control if the experiments are costly. So there are many reasons why statistically valid prospective validations are often not done. Yeah, in the end, what can happen is that you actually have quite a biased reporting. So you focus on cases where the method works, but you don't really have baseline controls. And that means in the end, quite often there's a focus on trivial wins. I mean, if you have 100 molecules you can test for a new method, I mean, it's tempting to pick one where you have some gut feeling or have some existing data already that the test will be positive for your particular new method. And that, of course, is then is biased in the way things get reported, and still doesn't allow us to say whether a method is truly better than another. So there are many problems with methods validation, is my point. Yeah. Another key point is that performance and data are related. So data is usually not sufficiently characterized to put performance into context. So chemical space is large, right? And if you use this part of chemical space to train your model or that part, you can get two completely different models. So you need to put the performance of your model into context of the data that was used to generate it. Yeah, that is very, very tricky. Comparative data sets are nearly always retrospective. Otherwise, you will need to spend quite a lot of money and effort to generate new prospective validation data. So you do use retrospective data sets, but that means they are limited in size, and we don't know the underlying distributions. You have many analogs in those data sets, so we don't really know which model is better. And if you do random splits, cross-validation, those things, you end up with similar distributions in your faults. What does it mean for the prediction of a new compound? That is quite often not so clear. Now, the bigger picture is, of course, that AI is where it is due in no small part due to human psychology. So what are bigger driving factors? Not always the science, of course. So hype brings you money and fame. Realism is boring. So basically, if you have a new method, of course, trying to put into the best light, okay, but that introduces a bias in science. There's fear of missing out. The others also do it, and beliefs often drive decisions. So that means that if one pharma company sees that 3 other pharma companies do one thing, there's a certain fear that they are left behind. And so this type of thinking, also on the company level, often drives decisions. Everyone needs a winner. So imagine a company invests lots of money into a particular method. And then, of course, you don't want to say that you wasted all that money. So you want to show successes, which then leads to the selection of, for example, some trivial examples. And of course, in particular when it comes to publications, you often have selective reporting of successes. It leads to everyone declaring victory. My method worked, that method worked as well. But in reality, there's no common baseline comparison. So no one knows what's actually going on, which method performs better. And that means it's difficult to really advance a field with little real comparisons of methods. So to summarize, so we need to analyze our data as we did for many years before, absolutely. And AI or deep learning in concrete terms is certainly a valuable tool in the toolbox, in particular if you have large data sets. But the real game changer for translation to patients probably will only come once we understand biology or biological data better and are able to generate it, encode it, and analyze it properly. Currently, a lot of computer science-driven approaches have been developed, and some are more applicable in drug discovery than others. Some of them are more tailored to the amounts of data you have for images and speech, for example. But the question is what translates to the drug discovery world, and this is still something we need to work on. And so real translation is necessary, but also better experimental design. And so that means that we generate the right data for a purpose, that we generate data that allows us to really benchmark models on a fair basis, and so on. When it comes to data, probably consortia on an even larger scale needed than before. So right now there are lots of efforts, at least in Europe, also in the US, for data sharing. But if you just share the data you have anyway, that might not be the right type of data. So we need to see how to share the effort of data generation, not just sharing what is there already, to really maximize the value of the data we generate, then for data-driven drug discovery in the future. So I'd like to thank you for listening. I hope you found the talk interesting and the whole event as well. Again, I would like to apologize that I cannot make it in person today, or at least live video at the right time. But please do contact me if you have any questions. Thank you, Renata, for organizing and for inviting me. I'm looking forward to hearing from you soon. Thanks a lot.
Okay, thank you for the participant attention. Now we get into the question and answer session. Okay, if anyone has any questions, please raise your hands, introduce your name, institution, and please state to whom this question is addressed. Question for Professor Bender will be answered in writing, and we will send Professor Bender's answer to you via your email. Ladies and gentlemen, we start question and answer session. Please deliver your question to our speakers. Okay, while waiting for live questions, I will read several questions from Zoom chat. And Prof. Enade and Dr. Maiwan, both of you have several questions. I will read first question for Pak Maiwan. The question is from Ignatius Andika Nugrahanto. I want to ask, is there any different pharmacophore model besides 3-point models like that triangle model, just like maybe 4-point model like rectangle? And then if there exist these models, which model is more stable? Thank you. Please, Dr. Maiwan, your comment.
Thank you, Pak Jeffrey. Also, Pak Ignatius. Previously, I don't really understand, but maybe I just can try to answer. Yes, of course, it's depend on the researcher to customize actually how many pharmacophore we want to generate. Sometimes there is some maximum value, the maximum number of pharmacophore, but we can customize. Actually, we can customize that. Please do the software to identify at least 4 pharmacophores, and also we can just play around that. If let's say you want to design the compound with that you think it's more stable with additional something like pharmacophore, yes, you can just customize that and instruct the software to generate for pharmacophore. Is if the software makes sense, so then it can be generated, that it could be active as well. That's all, I think.
Thank you, Pak Maiwan. Okay, the second questions for Pak Maiwan too,
Yes.
from Prof. Enade.
Wow. Okay, thanks.
Okay, I will read it, Pak Mai. Can we adjust the distance constraint in the ligands code?
Yeah.
Thank you.
Yeah, as I response in the chat, I'm not sure because so far I just use the demo version, so it's very limited. So I just use the default parameter. But I think yeah, is there any customization? How much constraint, distance constraint that we want to customize lah. I think so, but not sure because Ligensot is I don't have a license and just use a demo trial, which is very limited. We just do the default parameter. Thank you, Pak Jeff.
Thank you, Pak Mai. Pak Mai, this is for Pak Mai too, from Molvi Hanif.
Okay.
Okay, the first question is, What is the best protocol for evaluating in vitro new drugs? Example, PPI or calcium channel blocker or as a drug. Potential and efficacy using docking study.
Okay. What is the in vitro or in silico? What is
What is the best protocol for evaluating in vitro?
in
In
vitro?
vitro,
In
yes.
vitro,
In vitro new drug.
new drug for PPI.
Yes. Example like PPI or
Proton.
calcium channel blocker.
Unfortunately, I'm so sorry that I don't have any experience with this kind of in vitro assay using PPI or uh, any ion channel. Yeah. So I'm sorry, I cannot answer. My experience is to do in vitro assay for neuraminidis virus for influenza, and then dengue protease, and then matrix metalloproteinase for cancer and wound healing, and then histamine. Uh, what else? Acetylcholinesterase. What else? Alpha glucosidase. That's all. So I don't have any experience. Maybe, yeah, I can. I'm sure there is. Yeah, I'm sure that, but I just don't know because there is. I don't have any experience on this kind of in vitro assay. Thank you, Pak Jeff.
Okay, Pak Mai, this is still from Maulvi Hanif.
Yes.
Is it possible have difference of affinity or efficacy between structure compound in docking simulation with actual in vivo study? How to determine the accurate binding pose?
Yeah, of course. As actually, I often say, maybe also Pak Prof. Enade also agree that in silico not always 100% can confirm that our model designed by in silico should be or must be active by in vitro. There is so much factors, yeah. But at least by using in silico, we have been guided. We have any idea what kind of interaction, how much the binding affinity. But I cannot say that we just do docking for one compound, then we can expect that it will be active just by seeing that the energy binding is very good, although the interaction is very strong. Cannot. Yeah, we still have to do repetition, validation, compare with maybe do not design just one only and then more than one like pharmacophore to identify pharmacophore. It need at least 16 compounds. Yeah. So for me, maybe Prof. Inade can help also that it's not 100% can ensure, yeah, that the docking, something of pharmacophore and other will confirm or will ensure that it will be active or not active. But at least, for me, got the low active, it's I'm happy already. Let's say my docking is good, but in fact, when I tested it by in vitro, it got something like 30% inhibition or 30% activity, or the IC50 is like 100 micromolar. I'm happy already because at least my idea can get the activity. We cannot just use one compound, one docking that confirm that it will be active or not. That's all.
Can I add?
Yes.
An answer?
Yes.
Yes. Please. Prof.
Okay. Thank you very much. Then to answer the questions, and I would like to use this quote. Actually, all models are wrong, but some can be useful.
Yes.
Therefore, the retrospective validation plays an important role here in every virtual screening, ligand-based, pharmacophore-based, especially structure-based. We should perform this validation. And as Professor Andreas Bender also said, that sometimes it is just a game of number. But statistically speaking, the more compounds we use for validations, the more confidence we are of the predictions. That's my additional answer, Pak Jeffrey.
Yes. Thank you, Prof. Okay, for Pak Mai, still have additional answer again?
Okay.
Okay, thank you, Prof. N, and thank you, Pak Mai. Now we have a live question from Mr. Emmanuel Ramdhani. Please, Mr. Emmanuel Ramdhani, to deliver the questions.
Hello, good afternoon, atau good evening.
Yeah, good afternoon.
I would like to ask about if we have some protein target that has not been included in the database, can we find another alternative dataset for retrospective validation? It was the first question, and the second question, as I know, the database has the feature for making the decoy from our dataset. Is the decoy that generated from the database reliable enough for retrospective validation? Thank you.
Okay, untuk Mr. Dani, to whom these questions?
Prof.
Enade, I think.
Oh,
Prof.
Prof.
Enade.
Enade. Okay, thank you. Please,
Okay, thank
Prof,
you.
your comment.
Okay, thank you very much. If the dataset is not in the DUDA dataset, I would like to share screen if it is possible. So,
Okay, please.
okay, can you see my?
Oh yes.
So this is a DUDA dataset, yeah.
Yes.
So the targets are there. Unfortunately, I could not. Okay. How to change? The targets are there, but if you don't have the, what is it? You don't have the target there. We need to generate ourselves here.
Yes.
Okay. And you need to provide the DUDA dataset the smiles of the active compounds, your ligands. And the ligands can be seen in here. Yeah, you can find ligand in this data in this KMPL. Can you see my screen?
Yes.
Okay, you can try to find your compound, your active compound here. This is a curated database. So they try to find in journals, reputable journals, and then there is some of them, many of them, most of them are confirmed by lab tests. So you can check like this, what's the target? You can type the target there, and you will find how many compounds that have been tested and the activity. Some of them are not active. Many of them are active. And then you collect your own data set based on your own purpose, like perhaps using Lipinski rule to reduce the data set. Of if you don't reduce it more, using the fragment rules. And then you can grab the smiles. After we have the smiles, you can go to the DUDA data set and then generate the data set. That's the answer of the first questions. One of the techniques that I commonly use. Of course, there are many others, and there are some tools in Python to make decoys using, and then also some tools using KNIME. But I hardly use those kind of tools. The most common technique that I use is try to find the active compound in KMPL, and then go to the UDA to generate the decoy. That's for the first questions. And the second questions, yeah, can we rely on the decoy provided by UDA? My answer is that's the best we can have now.
Okay.
That's the best we can have now. actually the ideal if you have your own compounds, your own inactive compounds, but like Professor Andres Bender said, there are not many inactive compounds published. Yeah, it is difficult to compile inactive compounds for the decoy for our retrospective validation, unfortunately. But then, yeah, we can rely on this DUDA agent, DUDA dataset. Of course, there are some papers that also investigate the bias in this DUDA dataset. And then a good news actually, for about a month ago, I contact Professor Irwin, the one that create the DUDA dataset. They say discussing about the DUDA, and he said that now they are preparing another tools for ligand discovery called TLDR. So this is the tldr.docking.org. Yeah, he said that I should try to explore this. Then yeah, now I'm still exploring this, but I haven't used these resources for any research. That's for the second question. Is that sufficient?
Okay, can I ask another question?
Yes, please.
Question. So when I see the KMBL database, we can see that there are an active compound and inactive compound sometimes.
Yeah.
So can we combine the inactive compound with the decoy from the dude? I mean, when we generate from dude, we can obtain the decoy, and we combine it with the inactive from the KMBL.
The problem is perhaps the physical chemical property are not similar to the ones that we provide in ligands. So perhaps the inactive compounds is very different. So you do not need the filter screening to differentiate between inactive and the active compounds from the KMBL. Therefore, I do not recommend using those inactive compounds in KMBL if the physical chemical property are not similar. Yeah, there are tools that you can put a pool of those inactive compounds, and then you put a ligands, and there are tools to grab the compounds that have similar physical chemistry property, similar. So this is from the inactive. You should have the compounds that are similar to the ligands. Otherwise, you could not say that as decoys.
Okay. Good enough. Thank you very much.
Okay,
You're welcome.
Okay. Thank you, Prof. Enade. Prof. Enade, you still have two other questions. I will read first from Richa Mardianingrum. There are so many types of dude. Which file type to choose and what makes them different? Thanks.
It is a DUDA docking. Okay, I will go to the website. So for example, I will choose this IDRB2. If you go here, then there are many files. You are correct. I personally prefer to use the smile versions. There are why? Because the smile version is the unbiased form of the compound. You of course could get the SDF or MOL2, but then when you develop a protocol and you would like to test the compounds, you should have the same ligand preparations as prepared by the DUDA. Therefore, I personally prefer to use the smiles and then use our own protocols to prepare the ligands in the 3 dimension for the virtual screening. That's my answer. So just take this decoys final, and then also there is like a active final .ism, I don't know why they do not use the .smi, but this is, if you click here, you will find the list of smile compounds. And if you click here, if you click the one that with NM, you can also find the activity, the smiles, and then the activity in nanomolar. That's my answer.
Yes. Thank you, Prof. This is the second question from Abraham Oliver. If I may, I would like to know if the pipe leaf used the filter for higher prediction result, how the IPF exactly increase the EF and F value, Prof? And is there any standard of the EF and F value for acceptable virtual screening result of any novel drug compound? Thank you.
Okay, for the first questions, I will show you another screen. Hopefully now you are like a hacker now. So if this is the result of the the list of file resulted by the pipe leaf hypos. So I will show you one of them, the full IFP, full interaction fingerprint. You will get this kind of text. So this is a text file. So we could use the any text editor to filter the things that you would like to filter things that you would like to filter. For example, I would like to filter out compound that doesn't have interaction in this position, number 28. So I perform this script. Oh, sorry, number 29. So where are you? Yeah, this is, yeah. So if the data is 1, print it. So if it is 0, do not print it. Then you will lose the ligand ref number 10. So if it without the filter, you can see here ligand ref number 10 is there, but it doesn't have the interaction in this position. So using this simple script, you already remove the ligand number 9. When vice versa, if I want the post that do not have interactions, I just change this to 0, and you will just only get the ligand number 10. And we can make a simple script to systematically filter out one bit string by bit string. Yeah, unfortunately, that you must there's no user-friendly tools to do that. You must learn a little bit Unix shell scripting, like something like this, to do the filtering. Unfortunately, like awk and set, and yeah, this is actually only simple text editing. You can do you can perform it in Excel actually, but I don't know how to perform systematic filtering in Excel. That's the answer. Oh yeah, the question number 2, what was about the acceptable EF and F1, yeah, if I'm not mistaken. Is that correct, Pak Jeffrey?
Yes.
There are 2 questions. The first, how to do it, and then the second.
The second, well, Miss Prof.
About
the second, and is there any standard of the EF and F value for acceptable virtual screening?
There are no consensus actually, but I might suggest that if you develop a protocol of a retrospectively validated virtual screening protocol, you should have a better performance. So if this is structure based or ligand based, you should have the EF higher than the available ones. If not, yeah, I suggest that you can reach the EF or F1 better than the average. The average, I am not the average, there is in my slide actually, I think. Slide number, the slide will be delivered by the committee. Yeah, slide number 10. The average EF number in DUDA is 19.8, and the F1 value average of DUDA is 0.207. So there is not available filter screening protocols. Yeah, you can try to validate it and then reach EF and F1 better than those values. But if there's already available and you have a less number, yeah, you should provide why we should use your protocols. Like, yeah, this is a combination of blah blah blah, and it's much faster, etc. Just provide your arguments. Thank you very much.
Thank you, Prof.
Any other questions?
No, we don't have
Okay.
any question, Prof.
So,
Okay. Right. This is yes. Yeah. Okay, due to the time is up, we have to end this seminar. Thank you, Prof. Enade, Dr. Maiwan, and Prof. Bender for nice and inspiring presentation. Drug discovery is a challenging, time and fun-consuming process employing computational method able to reduce time and fun-consuming. Okay. We announce for participants who require a certificate, please deal with the rule of certificate. There are several. Subscribe channel YouTube Fakultas Farmasi Universitas Sanata Dharma. Participants must be present via Zoom chat or YouTube live chat with the name as registered with the committee. Participant must take a post test at the end of the event. Minimum post test score is 80. Participants must pay as 100,000 rupiah. And link post test. You can find this link post test to do your post test. Certificate and material from today's speaker, if speaker approved, will be emailed to participant email. Ladies and gentlemen, thank you for your active participation in this webinar. I hope you enjoy the webinar and able to participate again in the next our webinar on October 17, 2020 on pengembangan kosmetik bahan alam Indonesia dengan narasumber Ibu Dr. Dewi Setianingsih Apoteker, cantik dan segar dengan kosmetika tradisional, dan Ibu Kristofori Maria Ratnarini Nastiti, PhD Apoteker, dengan judul Formulasi dan Keamanan Kosmetik Bahan Alam Indonesia dengan moderator Ibu Dr. Sri Hartarti Yuliani Apoteker. Thank you for your participation and see you again.